All right. Well, my stopwatch has been running since yesterday. I stopped it after the last
Well, hopefully you can restart
16 hours and 20 minutes. Long presentation. Good morning, everyone. Welcome to the second day of the thesis presentation for IMBRC Core 2024. I am Olivier Gauthier from UVO. I will be in Brest. I will be chairing this first morning session. We have with us as members of the jury Nico Whiteberg from the University of Oviedo, Gonzalo Machado, also from the University of Oviedo, Patricia Pinto from the University of Algarve in Faro, and Nicolas Jegric from the University of Brest, also like me. So, as a reminder, students, you have 15 minutes to do your presentation, and then a 15-minute discussion will ensue with the jury members. When you come to the 13-minute mark, I will wave. 12-minute marks, I will wave 3 fingers in the air, signifying that you have 3 minutes left. You deal with that. If I get up, that means you have been grossly over time and you need to wrap it up. But that's not happened in the past day, and so I'm pretty confident it's not going to happen today either. So it is now 9:30, so we can start. Cassidy, whenever you're ready.
Okay. Hi everyone. My name is Cassidy, and I completed my thesis in the UK at the University of Plymouth under the supervision of Dr. Emma Sheehan, and I'm excited to tell you today about long-term biodiversity recovery in the Lime Bay Marine Protected Area. So to start, bottom trawling is one of the most destructive fishing practices. It has the highest discard rate of all fishing gears, and it contributes to almost half of the total annual discards by global fisheries. And the photo in the background is a real photo. It shows the extent of trawling. It was taken in the Gulf of Mexico in 1999 by one of NASA's satellites, and it shows that the sediment trails caused by trawlers can be seen even from space. So because of the extent of how trawling affects the marine environment and marine life, it was really surprising to me to learn that in the UK only 10% of MPAs are protected from bottom trawling. One of those MPAs, though, is in Lime Bay, and that's the focus of my research today. So you can see the orange rectangle on the map that shows it's in the southwest of England. It is home to all these reef habitats and over 300 different species, including ones that are threatened, rare, and also commercially valuable. So for example, pink sea fans, which are slow-growing cold water corals. We have king scallops, which are an important commercial species, and then we also have Ross coral, which are really vulnerable to bottom trawling and dredging. They're actually not a coral, though, they're a bryozoan. So despite being a hotspot for biodiversity, Lime Bay has experienced an intense history of heavy fishing pressure, particularly scallop dredging. So scallop dredging is when there's a heavy metal frame that digs into the sediment and the benthos, and it destroys, it just like drags over the benthos and completely destroys it. So public outcry from conservation groups, scientists, and also fishermen in the area led to the establishment of the Lime Bay Marine Protected Area in 2008. When it was established, it was the UK's first and largest MPA that adopted a whole site approach. So that's when the whole area is protected, not just specific features like just the reefs. So 206 square kilometers were protected from bottom toad fishing. My objective of my thesis is to look at is recovery occurring in Lime Bay, and I did that through looking at abundance, taxa-richness, and also assemblage composition. My hypotheses were first that abundance and taxa-richness would increase in the protected areas over time compared to the open controls, and the assemblage composition in protected areas would become less similar to those in the open controls. The survey design for the sampling in Lime Bay consisted of 3 different treatments and then 1 control. So the previous voluntary closures were areas that were actually voluntarily closed by fishermen in 2001, and then they added more areas in 2006. The statutory instrument is just another word for the legislation that created the NPA, and that was in 2008. So these first 2, the blue and purple, they're the most protected treatments. They adopt a whole site approach. And then in 2011, a special area of conservation was established in and around the marine protected area, and that was feature-based protection, so just the reefs in the special area of conservation were protected. So bottom toad fishing actually can happen around the reefs, just not on the reefs. And then open control, there's no protection, and the sites are located outside of the marine protected area. This is a map of the sampling design, just for visual purposes, but the whole light blue shape is the special area of conservation, and then we have the darker blue outline is the statutory instrument where the whole site protection is implemented, and then the gray shapes are the previous voluntary closures. And all the treatments are color-coded. There's 6 sites per treatment and 3 replicates per site. So those sites were sampled from 2009 to 2024 using baited remote underwater video, or BRUV. So the BRUVs consist of a metal frame which have 2 weights attached. They have a camera, which we use GoPros, LED light, and then they have a pole extending from the side of the BRUV, and that's filled with mackerel. And yeah, you can see the BRUVs in this photo here. So I used a combination of data from footage that was previously analyzed while also analyzing my own footage from 2024. The BRUVs basically get deployed off the side of the boat, and they're left for a minimum of 45 minutes to film the surrounding marine life. And then we get them back and look at the footage. So for the footage that I analyzed, I counted the maximum number of individuals for each taxa within 1 minute of the video for 30 consecutive minutes, and I identified species to either species level or lowest possible taxonomic rank. In the photos here, you can see some of the screenshots that I grabbed from the footage that I looked at. We have conger eels, scallop, and then pelican. So that was step 1, which was video analysis and species identification. Step 2 was to combine data from previous years with the data that I newly analyzed, and then step 3 was doing statistics and data visualization. So looking at this graph of abundance, we have 4 transformed abundance per site plus or minus standard error on the y-axis, and then we have year on the x-axis. And when looking at initial versus final 3-year averages, all treatments increased. And when running a linear mixed model, I found a significant effect of year, but a non-significant effect of treatment, suggesting that all treatments increase over time, but differences between treatments were not significant. Also, looking at taxa richness, all treatments increased when comparing initial and final year averages from 2009 to 2024. And then looking at a generalized linear mixed model, year had a significant effect and treatment had a non-significant effect, similar to abundance. When looking at abundance and taxa richness side by side, you can see there are noticeable decreases. So in pink, it's 2014. That has been attributed to severe storms in Lime Bay that happened in the winter of 2013, between like 2015 and 2014 sampling periods. And those storms like completely destroyed the benthos, and in the following year, in 2014, we saw a lot less abundance and taxa richness. And then in 2021, in orange, there was also a decrease. And one theory for that is that that was the year following the initial year of the pandemic, and there might have been some increased fishing pressure that caused that. So when looking at assemblage composition differed significantly among treatments and years, as revealed by a significant per-meter test. And an NMDS plot is a good visualization of this, where each point is a year, and so you can kind of follow the trajectory of assemblage composition through the years. And we see a separation between the previous voluntary closures in blue and the statutory instrument in purple. Those two are kind of separated from the SA and the OC assemblages. And that kind of suggests that the whole site protection in PVC and SI results in a different assemblage composition than the feature-based protection in SA and no protection in OC. And this aligns with previous research as well, because it's been shown that whole site protection offers higher increases in abundance and also in functional redundancy. I also completed a SIMPER analysis, which stands for similarity percentages. And this analysis is basically telling us which species contribute the most to differences between different groups. And so the panels here are comparisons between the controls and PVC first, SI, and then SA. So year is on the x-axis, and then each row represents a different taxa. And the mean contribution is here. The yellow-orange colors represent a higher mean contribution, while the purple and pink kind of colors represent a lower mean contribution. Across all comparisons, the taxa that contributed the most to differences between control and treatment groups were gold cine wrasse, pouting, hermit crabs, and a species of brittle star. So when we look closer at these four taxa, it's easier to see the differences between treatments and abundance. So here we have year on the x-axis and abundance per site on the y-axis for each of the taxa. Gold cine wrasse and pouting have the highest abundance in PVC and SI, and they are known to inhabit rocky substrates and reef habitats, which may explain their higher abundance in PVC and SI, because those are the treatments that have been protected for the longest and are the most protected. Hermit crabs have the highest abundance in SA and OC, and there was a study from the southwest coast of England that suggested that abundance may increase with greater fishing disturbance because of increased availability of food from bottom tow fishing for the hermit crabs, as they're detritivores and scavengers. Brittle stars show more variable trends, and there was one study that showed that the relationship between fishing activity and this one species of sea star kind of represented a potential trade-off between the increased food availability from discarded bycatch and mortality associated with trawling. So that may kind of explain this. So some of the limitations in this study were the timing of sampling, and they always tried to sample during the summer months, but it kind of varied in terms of the exact days and the exact months, mostly because of weather, and so that could have introduced some unaccounted for variation. Then conditions during sampling. So depending on mostly weather and sometimes algal blooms, the visibility in different videos would differ. And I even saw that when I was looking at videos myself. Sometimes things would be really easy to see, and sometimes you would have difficulty identifying species. So that also could have caused some variation. Observer bias just refers to the fact that a lot of analysts have contributed to this data set over time with the analysis of footage, and we all have different skills of taxonomic identification, so that's a bias. And lastly, different taxa respond differently. So what I mean by that is that depending on whether they're commercial species, non-commercial species, or maybe vertebrates versus invertebrates, like different groups of taxa respond differently to different pressures. And I looked at total abundance and taxa richness, but different groups do respond differently. So recommendations for future research. I would recommend, based on the results of this study, continued monitoring in Lime Bay to contribute to this extremely valuable data set, and also the establishment of whole site protection in the special area of conservation instead of just feature-based protection. In the future, I would recommend investigating ongoing fishing activity that's still happening within the NPA, so that can inform catch limits for species that are caught using traps and static gear, and then also looking into the actual pressure of static gear in the NPA, so that we can understand how that's affecting ecosystem. So in conclusion, abundance and tax richness significantly increased across all treatments over time, and they showed higher increases in PVC and SI sites, but they did not differ significantly between treatments. PVC and SI assemblages were distinct from OC and SA, and species contributing most strongly to assemblage differences were Fulton wrasse, pouting, hermit crabs, and brittle stars. And overall, I think the study shows importance for continued monitoring in the NPA and how that can help us inform conservation decisions. These are my references, and I just want to thank the IMBRC Consortium and my supervisor Emma and the team at the University of Plymouth, and then all my friends and family as well. Thank you.
Thank you, Cassidy, for this very interesting presentation and signal the perfect use of the time you had. So, congrats for that. So, jury members, we'll try to stick to 3 minutes each, and then that gives us a little bit of wiggle room. So, who would like to go first? Nico, please. Yeah.
Okay, thank you. Cassidy, very nice presentation. I really enjoyed how you framed the presentation of your work. It's obvious that you work quite a lot. It's very apparent in the presentation. Also, I like a lot the right combination of just the right amount of text and beautiful images and maps. Very nice. I have a couple of questions. The first one is, have you tried to look at the trends in time for entire functional groups and not only some species? Because why did you select these species? Because they were the most common, or?
So the species that I looked at were the species that contributed the most to differences in treatment groups. So looking at the SIMPER analysis, they were just the top species that had the highest mean contribution, and I just wanted to look closer at those species to see like which treatments they were more abundant in, and why I could explain some of that. But that is a good point, because functional groups do tell like a different story, and if I did have more time, I would want to do that as well, looking at like different either scavengers or just different.
The point is that you have some theoretical explanations on why did you observe those patterns for each of those species. You can test that if you put a similar species there, a functional group. It should follow the same trend. If not, maybe your hypotheses are not correct and you have to change. So, the other thing is, do you have any idea of how can you correct a little bit for the time of the year the sampling was done?
If you maybe include like some of the date in the analysis, like the day year, like including,
But as a covariate,
Yeah.
I think, let's see if that has an effect or not. Maybe you can help me. Yeah, nothing more from my side. Thank you very much.
Thank you so much.
Thank you, Nico. So maybe we go down the line.
Okay, yeah. So, great talk. I really enjoyed it. So it's not really my topic, but I really appreciate the way that you engaged the audience so well done. Also, the figures were quite clear with the message that you are giving to us. So well done. So, and I mean, you already acknowledged some of the limitations of the study. Of course, this is a long-term study, so you need to come with data from other researchers.
Right.
But have you do any attempt to test the repeatability of some of the years that you have? So which is the effect of the observer or of the researcher when you are doing the analyzing the videos?
Like, are you asking how to make sure that's consistent, like the way that things are
Yeah,
being
somehow,
analyzed?
yeah.
So that is something that I was discussing with the team there, and it is, I think, an issue that hasn't really been addressed yet because when I came in to analyze the videos, like someone was teaching me how to do it, and like there was kind of just like word of mouth was how they were teaching me how to do it, and I actually ended up writing like a standard operating procedure because there was none there already. Just because the way that I was analyzing the videos, like sometimes I would go through and watch like the same species throughout the whole video and just try to, because they move around and try to figure out how many there were in each frame. But then sometimes I would do just 1 minute and look at all the species. And so I think that really depends, but it is something that, you know, if you had more time, maybe you could go back and try to like QC, do some like quality control for previously analyzed footage. But just because of time limitations, like I just analyzed 2024 and then added that to the data that had already been analyzed.
And correct me if I'm wrong, but you say that for each point you have triplicates, no? Or
Yes.
you have, how
Each side.
different do they were? You have some
Um,
estimate of that, so
I don't have like a quantitative answer for that, but when I was watching the videos, they were quite similar in like, because they're only 100 meters away from each other, and I actually got to go out on the boat and like do some of the sampling. So they're quite close relative to each other. So you would often see like similar things because we deploy them at this exact same time on the boat. So yeah, I don't have like a quantitative answer of how like similar.
okay, just the last one.
Yeah.
Is there any attempt to try to do some automation of the video analysis? Because I know when you have one target species, it's much easier. So in your case, it's quite complex, of course. But
Yeah.
is there any attempt to try to reduce it? Because type is quite time consuming, isn't it?
It is.
So,
It's very time consuming.
You know much better than I. So.
Yeah. So not that I know of, like they've been doing this monitoring for a long time and it's always just been manual, using like someone, a student or someone else. But there actually was someone at the University of Plymouth who was working on that exact thing, like using AI to try to automate that species identification. I just think it's like so hard because even as like a person, it's hard for me to identify species. Like I have to keep replaying, and like sometimes the visibility is you can't see it properly, and like I just don't know if the technology is there yet. But that would be great.
Thank you very much. Great job.
Thank you, Gonzalo. Thank you, Sue.
Congratulations, it was very clear and very interesting. So, and I also don't have many questions because either they were asked already, it was about the species or the time of the year, or also because it was clear. So, and to understand what steps did you have the hypothesis to engage, not only the videos, but did you go on the boats and the recording and all that?
Yeah,
Can you
so
elaborate on that?
we were supposed to start in June, and I was supposed to go out with the team and sample, but as you know, the weather in the UK is very sporadic, so it actually got canceled for a few times, but I did end up going out for 2 days to start the 2026 monitoring because it just got funding for that, and it was really helpful to see how the brubs are actually set up and deployed, and there are actually a lot more things that go into it than you would expect, and like having the GoPros on and making sure the lights are on and just like having all the sites organized and everything, but it was pretty nice to see that.
And then the rest data analysis of the previous. one.
Yeah, I did that
Okay.
before.
And now a curiosity, is there any citizen sciences or education projects around this? And if you had the chance or not, if interacting with this?
So there is outreach that goes on with the team that I was working with at the University of Plymouth. They do host webinars, and they have a really strong relationship with the fishermen in the area. So we work really closely with the fishermen, and there's been studies after the NPA was established to figure out their perspectives on the NPA and whether it's something that they wanted and whether they like seeing anecdotal evidence of like whether they see more lobsters or crabs or whelks. And so I think there's a really good like relationship between the science and the actual stakeholders.
Which is very important also for education, for them to change
Yeah.
habits, release of videos, all that.
Definitely. And like this all started because fishermen were concerned about the dredging and the trawling that was going on inside the NPA, and that's how it started with the previous voluntary closures. And then the fishermen were the ones who were kind of like supporting the establishment of the NPA.
Because now we also have an MPA in the Algarve.
Oh, nice.
That
And like in
there are
Maria.
several groups that are getting engaged with the fishermen, also for education of habits
Right.
and for environmental protection and all that awareness. That's us clear.
Thank you, Cassidy. I also really appreciate the clarity of your presentation, as it was said. I've got one question about the possible other variables that you might have analyzed, because you are only focusing on the biodiversity aspects and the community composition. And I was wondering if you tried to see if you could explain that by some environmental variable, perhaps.
So I didn't look into that. Are you thinking like depth or
For instance,
yeah, okay. So we did try, all the sites were selected like before the monitoring had even started in 2008. And they selected the depths, or they selected the sites based on depth and then habitat substrate and also fishing pressure historically. And so I think they did try to select the sites with like keeping consistency in mind. I didn't look into like whether depth was a factor, but I could have in the analysis, like the exact depths of the site sampled. But yeah, I do think they try to keep it like quick.
okay, yeah, because that was something that worried me a little bit when you see those differences in composition. You also have a pretty clear gradient, at least in terms of distance from the coast, where the most protected area are closer from the coast, and the little less protected area are further away. And I was wondering if that could explain the differences you see in terms of community composition more than the protection itself, perhaps. Do you have an idea?
I think that so on the map, the sites do look like pretty far away from each other, but in reality, I think they're actually quite like closer than they look on the map. And I think maybe the spatial aspect of it may be a factor, and like the fact that the SA sites are closer to the OC, and maybe that's why the assemblage composition is the same, or is similar. But I think that in terms of like distance from coast, I don't think that would be like one of the main factors in looking at like assemblage composition differences.
Okay.
So left time.
Okay. I had another one, speaking specifically of like trying to decipher a little bit this story about protection. Do you have any data, I guess not from Grove, but do you have any data before the protection?
No, so there's actually no like technically like a proper baseline because they didn't start monitoring until 2009, and the NPA was established in 2008. So like the first year that we have is when the NPA was already protected. So that is also kind of a factor that we have to take into consideration. But before that, I think it would just be like anecdotal evidence from fishermen and their perspectives.
Okay. And have you been able to look into that a little bit, or just like even if it's only tentative? I mean, if it's anecdotal.
Well, okay. All I can say about like before the NPA is that the anecdotal evidence from fishermen, they did see like the benthos being destroyed by the scallop dredging and the trawling, and I think that's why they were so vocal about like wanting to create these protected areas. And after the establishment of the NPA, like they've seen more of like the sponges and like more of like increases in abundance and everything. But other than that,
Thank you. Anybody has a quick question to finish? I do. I was wondering if in your model you tested the ear and the treatment effect. Did you explore at least if there was an interaction between the two?
yes, I did look at if there was an interaction, and there was no interaction between the two. I also used sight as a random factor just to have that in the model, but there was only a significant effect of ear.
Thank you very much. Done.
Thank you. Can I just see how this changes?
That one's blind.
Oh, okay. Thank you so much.
Angelina, whenever you are ready.
Thank you. Good morning. Today I will be presenting my master thesis defense entitled Evolution of Temperature and Nutrients at the Southern Portuguese Margin for the Last Millennium with the following structure. So to start, it is important to understand why paleoceanography is useful. Paleoceanography allows us to reconstruct past ocean conditions using natural archives preserved through time. This can help us understand past ocean variability, including how variables such as nutrients and temperature have varied in the past. And this is very important to help us understand how they can also change in the future. This is especially relevant for marine ecosystems and living resources that can be strongly affected by climate and oceanographic variability. This is an image that shows the climate variability in the northern hemisphere for the past 11,000 years. And is to show that I will focus only on the last part of this variability, the last millennium, that included two major periods. The medieval warm period that was characterized by warmer conditions, and the Little Ice Age that was characterized by colder periods. To reconstruct these past ocean conditions, I used different marine archives, specifically the ones presented here, deep sea corals, especially these two species. I used the genera of Ovigerina for the benthic foraminifera, and these two specific species of foraminifera for the planktonic foraminifera, Buloides and Inflatum. Moving on to why these marine carbonate archives are important. So deep sea corals and foraminifera, they incorporate in their skeletons or shells, in their carbonate skeletons or shells, chemical signals from the surrounding seawater at the time of calcification. So, by measuring their elemental ratios, we can then have an idea of how the conditions of the chemical conditions of the seawater were at the time of calcification and skeleton formation. So, by calculating these elemental ratios, and then they can be used as a proxy for example, nutrient and temperature variability. So, based on this, my three research questions are: how did this temperature vary at the southern Portuguese margin for the last millennium? Also, how have nutrient and exported related conditions varied based on baryocalcium ratio? And how are these variations expressed across different archives and climatic periods? So, let's see. My study area was the southern Portuguese margin, especially the Algarve region, in those 2 points that you can see here, the 3 and the 7 point. Sediment samples for foraminifera were collected from the two stations, but for samples for deep sea corals, they were only collected from station 7 due to opportunistic sampling. They were collected using box scores, and this region is very important in terms of why it was being studied, because it, contrary to the coast of Portugal, to the west coast, it has seasonal and weak upwelling, water mass variability, and it can be influenced by river inputs, especially from the Guadiana River. So, in terms of my methodology, first for the foraminifera, they were the sediment cores were freeze-dried, wet sieved. Then we did the picking and identification for the different specimens. They were then crushed, chemically cleaned, and then prepared for analysis using ICP-MS. And in my case, I had to use ICP-MS and ISO-POS machines. For the part of the deep sea corals, it's relatively easier. They are just washed. Then they are mechanically cleaned using a Dremel tool to remove most of the surface contaminants and organic matter. They are then crushed using an agate mortar and pestle, chemically cleaned, and then also measured using ICP-MS or ICP-OS. Follow that, we get the values for the elemental ratios, and then we use those values through all of these equations to get an approximation of temperature values. For foraminifera, we use the ratio of magnesium and calcium with species-dependent formulas and equations. And for deep sea corals, we use the ratio between iridium and magnesium as a proxy for temperature. At this point of my study, we use the baryocalcium as interpreted qualitatively to assess relative changes in nutrient and exported related conditions. But we can do, and I will do it after, some calculations to just get the baryocalcium in the actual seawater, because this baryocalcium is the one measured from the actual foraminifera and corals. Aluminium, iron, and magnesium was also checked as contamination indicators in my analysis, and IRSD values from the machine and tokisfens were used as quality control and outliers groups. Now, moving on to my preliminary results and discussion, I will first focus on the range of values and then on the temporal trends seen for each archive. So first, an overview of the temporal values for all of my archives. We have beloids, inflata, vigirina, and deep sea corals, and what we can already see is that the planktonic species, which are beloids and inflata, record the warmest values, contrary to the bottom water archives that are the vigirina, our benthic species, and our deep sea corals that already represent lower values. So before looking at any temporal trends, we can already see a difference between upper water and bottom water records. From the values from baryocalcium, we can see that beloids, vigirina, and deep sea corals have very similar values between itself, but inflata shows the highest and widest range of values, ranging from 2.3 to 53 micromoles per mole, and I will then explain why this big difference between the species happens. Moving on to the graph that shows the foraminifera temperature values through age. This is done by an age model that was passed to me. I didn't do the age model myself, but it basically connects the sediment depth sediment with the age of the sediment itself. So looking at that, I basically have the stations 3 and 7 distinct. The station 7, oh, no, sorry. The station 7 are the ones here and the stations 3 are the ones here. I didn't use them together at this point of my presentation, but for after results I will, because they were technically taken from different stations, so they should be interpreted separately. But with the age model, we basically realized that one of the stations, the top core, it's actually from the middle of the Middle Ice Age, not recent, so this needs to be taken into account. But basically, we have the Vigirina in green, the Inflata in red, and the Beloids in blue. What this is showing, firstly, is that they all represent different ecological habitats in the water column. As it was predictive, because Vigirina is a benthic species and these two are planktonic species, but we know that Beloids normally lives in the upper water column of around 50 to 100 meters, while Inflata, for this reason specifically, it ranges to normally 250 meters. So it is normal that they're representing stratification and different water columns. Beloidish, the one in blue, it has higher values. All of them have higher values in the medieval climate anomaly, and then a peak when it drops to the Little Ice Age, which is coincident with this being obviously colder intervals. The Beloidish also represents here kind of like a shift at around 1600, which is coincident with previous papers, especially Salvado 2005, that basically says that around this time there is a difference between the Lea, with the first part of the Lea representing more warmer conditions and the second part colder conditions. The inflata presents a more stable running average, but still has higher values during the MSA. The Vigerino represents a different pattern that we shouldn't analyze as differences in surface warming per se, but differences in bottom water conditions, obviously. All of these periods in the most recent part, even though it needs to be addressed very carefully because we have a lot fewer samples, for the sedimentation rate that we have, that is 30 centimeters per millennial, even this region showing already an increase, it can show and can be already presenting modern climate warming. In terms of the foraminifera, as I told you, they have very different patterns from the inflata that I had to separate from the beloides and Vigerina, especially because different papers have suggested that inflata, because it's a non-spinous foraminifera, it tends to calcify within baryo-enriched organic aggregates, such as marine snow, which can basically alter their baryocalcium values that are not reflecting just the seawater, but their ecological and vital effect patterns. The beloides and the Vigerina have a very different pattern because they have higher values during the LIA and lower values during the MSA, which is contrary to what inflata is saying. So they need to be addressed as they're reflecting other patterns. And what I discussed is that basically they're probably reflecting river-driven nutrient inputs, especially from different flooding events that occurred at this time. Specifically, this high period here can be, and it's mostly related to flooding events and from river inputs from the Guadiana River. So, moving on to the bottom water archives, the color in green is representing the Vigerina, and the color in pink is the deep sea corals, because they both represent bottom water archives. And the part in blue, it represents the organic carbon to see if we have a connection between organic carbon and baryocalcium, because that's what you would expect. So in terms of temperature, they have very similar patterns, which is that they have a rise until 1,200, and at the beginning of and the onset with LIA, they have a decrease, which it's good that it has a similar and more coherent pattern, because it means that they're both undergoing the same bottom water influences from the layers of the water column. But looking at the baryocalcium and connecting with the organic carbon, the Vigerina has really small inflations that it's hard to see here, but it has small inflations that are partially coincident with the organic carbon. But the corals are relatively stable for most of the period and only have a decrease on the onset with LIA, which basically can demonstrate that, in a way, the baryocalcium and the organic carbon, it's more, the Vigerinas are more influenced, and it can have a higher impact in them than the coral. They're not that affected by the local sedimentary organic carbon as the Vigerinas are. In terms of future research, one thing that is really important is the independent dating using isotopes of the corals itself, because we actually have no way to be certain that the corals are the age that we are saying they are just because they are in that layer of sediment. In terms of another thing that we could do is the higher resolution sampling. I did my resolution was from 2 centimeters, but it would be amazing if we could do 1 centimeter each, so that we have a better understanding of the water column. One thing that we can also do is a longer and deeper sediment core selection. Our cores had around 20 centimeters and one until 25 centimeters, but it would be amazing if it would be until, for example, 30 centimeters. Also, one thing that we could do is improve the multi-archive comparison, and finally, a standardized ICP reanalysis of the complete sample set. Just doing ICP analysis, because I will explain after, I had to do ICP-MS and ICP-OS, and I will have to redo a reanalysis of the complete sample set just doing ICP-MS. So to conclude and returning to my research questions, firstly, in terms of temperatures, we could see that there was warmer during the MSA, and then a cooling transition during the LIA, with LIA being an interval that had variability within itself. And in terms of the most recent interval, we can already see an increase, which may represent some idea of modern climate warming. Second, we had a variable nutrient and export signal that could be linked to a lot of factors, but specifically from my region, precipitation, flooding events, and river inputs. And third, we had different archives that recorded different water column layers in depth. Specifically, the beloids seem to be more related to river influence and productivity. The inflatus seems to be recording a different, deeper subsurface layer. The uvigerinas, bottom water conditions, according and also with the corals, but specifically for uvigerinas, remineralization and nutrient export near the seafloor. And all of this information show us that the changes that occur in terms of temperature and nutrients in the southern Portuguese margin for this region were not homogeneous during this millennial, but changed according to a different sort of factors, including the climate period, the species ecology, the archive-specific responses, and even the species itself. Thank you. And this is my references, and thank you. Any questions?
Thank you, Angelina. Very well done. Perfect use of your time also. So, go over to the jury. Maybe we start with somebody else. Nico, if it's okay with you. Who wants to go?
It's all right, I can start.
Yeah. All right.
So thanks for the presentation. Really nice presentation, by the way. So when you say preliminary result, it's because
Yeah.
you are planning to present the second version?
Yes.
Okay.
So I had a problem in my lab. We needed to measure the results using the ICP-MS, but those types of instruments need to be used in deep, clean lab conditions. And we had a problem since December. We couldn't make the lab have those conditions, and we only made it work at the beginning of June.
Yeah, things happen. I mean,
That's
it's nice as you mentioned, but yeah, so things happen. That's
Yes.
So, by the way, have you thought, or do you have the chance to get the opportunity to analyze other sort of data from a near region or so some normally I really like when you are doing an experiment or research to have some control condition or something that you can compare with. Do you have the opportunity? I mean, this is depending the availability of sample, of course, but
It's really hard because actually most of the sampling that was done, especially in my lab that I worked, it's done. I can show you, but it's done more on the west coast of Portugal, like I said. Here. It's mostly done like in this region. There's not a lot of sampling points specifically in the southern region, especially near this point that I worked, because this region is strongly affected by upwelling. But my region, the influence that it has from upwelling it's weak. So most of the studies are done here or even on the coast and not in this specific part. So that's why they also want me to do this data specifically here, because there's not a lot of information. And the only information that we have from different papers that study the oceanographic variability here is that the upwelling is weak, but they have information that specifically for this region, it has been increasing through the years. So they also wanted this region specifically for that part.
And the last question, what about the models? I mean, you clearly explained that, I mean, you need extra data to really trust the aging models. So, but those models were already applied for samples in that region? So, because depending on the position, many other factors that might affect the way that you estimate it. So,
so basically, the age model itself, it's from information that will be eventually published. It hasn't been published yet, but it basically it was done using radiocarbon and lead 14 to get the information, and it was done at the time of when this sampling was done. Because one thing that I needed to say was this was done during the Eurofleet Impact cruise. This was in 2012, but I'm analyzing those samples that they have now because they didn't have anyone to analyze those samples until this point. So the age model was done at that time from a researcher there, and we tried to see if the age model was correct, because basically I have two stations, and this specific station, as I said, the top core is from the middle of the ice age, And my advisors, when they saw this at the first time, they didn't believe it was correct. So we we tried to see like if it makes sense or not. But then when I analyzed the data from the corals and everything together, it had a correspondence. So we believe that the age model is correct, and we use it because of that.
Thank you very much. Thank you, Dr. Nicola. Nicola.
Yeah, I've got a very kind of broad question because I'm not a paleoecologist by any means, and I was wondering if you could explain quickly, if possible, all of the specific element ratio related to the temperature.
I can. Here. Okay, so basically, there are lots of elemental ratios that you can use as proxy, but I use these ones in specifically because they are the ones that normally are more studied and can provide them the better results. But we use this because when the corals and the foraminifera are forming their skeleton shells, they are incorporating those elemental ratios from the seawater at time of calcification. So they change the amount of magnesium and calcium that they are, in this case magnesium, they're incorporating. It will change through time. So they will incorporate more or less. And then these equations are already done by each species or specimen, and they do calibrations to try and see if they have a linear or exponential correlation between them, and then I use them to calculate an approximation of temperature. I don't know if this was
Yes.
Yeah.
Yeah. I'm kind of curious about how do they calibrate those equations?
Who?
How do they do?
How do they calibrate
Yes.
these equations? Well, they do tests with the specific species of foraminifera, and they also use the ICP-MS machines, and they try to see, okay, this calibration was almost perfect, and then we do another calibration, because even we in the lab, we have to do calibrations before we even measured
Oh yeah,
the
for
animal.
the measurement.
Yeah.
So, but I was thinking of the temperature. Is it because they need to do those calibrations? They need an independent source for temperature.
They need to.
An independent source for temperature, probably.
Yeah.
So that they can say, oh, this is a relationship, and then give it to you so that you can use it. But for instance, for where does that temperature come?
I think they just take it from. Because, for example, for the paper that I saw from the corals itself, they just assumed the temperature for the specific region that they were measuring.
Okay.
So I don't know, like WOA, WOA 23 NOAA. They.
Or like for modern.
Yeah, exactly. I can't remember the actual name, but
Okay.
it's WOA, and they take the information from that temperature itself, and then they calibrate to.
Okay, so it's specifically calibrated with modern.
Yeah. It is calibrated.
Okay.
But it's also sometimes it's not only species dependent, but it's locally dependent. So it depends. For example, if it's corals from the Caribbeans, or you see, it will have an impact. But the calibrations are much more specialized for the foraminifera than they are for the corals, because it's way harder to do it on the corals. Because, for example, the magnesium and calcium can be measured in an ICP-OS and not in an ICP-MS. But measuring lithium in an ICP-OS, it's very hard, because lithium in corals and in foraminifera, but especially in foraminifera, it's really like a really small number. So it's very hard to measure it. You need a really good machine to be able to measure it.
Okay, well, congratulations for the presentation. I really enjoyed that. Nice pictures, drawings. I really like that a lot. And can you go back to your map? Because it's true that you are not You are just at the border of the upwelling region, right?
Yes, technically, the papers that I read, they say that when the upwelling is strong enough, it can reach
Exactly.
this region. So I'm exactly at the border where the upwelling is weak, but can already be happening and be seen.
Yeah. And do you think there were other times in the past that that happened? Can you tell that with your data?
I think so, and that's what I want to explore more because basically Bloidish, the most surface planktonic foraminifera, normally it's the foraminifera that people use to connect with upwelling because normally it's the one that because it exists at the surface level basically, it can be connected to upwelling, and there's a lot of papers that connect Bloidish with upwelling. The problem is because my area is technically an area of weak upwelling, my advisors, they're not completely certain that this Bloides specifically is responding to upwelling and not river-driven productivity. Because basically the Bloides, she vertically migrates in the water column based on the area that has more productivity because she goes wherever she has food availability. So that's why it's hard to interpret from my data if the Beloid will be responding to either upwelling or if she will be responding to river productivity.
Yes, but I wouldn't discard upwelling because
Yeah,
you are,
I
you
also
are,
wouldn't discard it.
you are downstream. The current is going
Yeah.
from the north to the south, so it takes some time for the phytoplankton to
To-
develop after an upwelling event, and in that time you move south because of the current to your area.
Mhm.
So you can have upwelling bloom events, phytoplankton bloom events in your area that were produced by upwelling upstream. You know what I mean? So
Yeah, I agree. I also when I received the data and I was talking to my advisor because she has a paper that she published herself where you have a map and you can see where the Beloides sampling points and were and how the upwelling is. And basically in that map, it's basically the Beloides is representing upwelling in all of the areas except for that one. So that's why she's also indecisive if the Beloid will be representing upwelling or river productivity. But I think it can even be representing both at some point.
oh,
So it needs.
yeah. My last question. Or
Maybe if we have time after Patricia.
Okay, sure.
Okay.
Or you can, I only have one, so yes.
Welcome.
Congratulations, Angelina. This is not my area, but I could follow very easily, which means that you understand very well and could pass and explain all the limitations and all that. I work at different level, molecular biology and biotechnology engineering, which leads me to technological also questions. So you said that we will need to repeat by the different ICP, MES, or the other one.
Yeah.
What are the technical differences? What will be the advantages, and how could it improve the results?
Yeah. So because of the problem that we had in time constraint, and because I wanted some data to be shown in this presentation, we actually sent half of our samples to China because that's where my advisor had a researcher that could do it for free. But the problem in China is that it's true. It's true. It's true. But the instrument that we have in China is an ICP-OS, and basically the ICP-MS it measures the different atoms by the weight of the different atoms in the isotopes. But the ICP-OS it measures the excited electrons. It measures how much of the electrons were excited. But basically, because they measured in different ways, they still have the same results. But the problem is the ICP-OS has a lower detection limit range. So if you're trying to measure really small isotopes, and especially for elements such as lithium, it's really hard because it doesn't have the same power as an ICP-MS that can measure it without no problem. So that's why I said that we need to do a reanalysis of the whole samples but using an ICP-MS so that we can be certain and that we can have the same precision even because the other one can't.
I don't know if you consulted that CISMAR, that is ICP-MES, and that is MPAAS. Which will be different, I think.
My advisor consulted all of the options, even in Portugal. But the problem is, even if it's an ICP-MS, it doesn't mean it can measure our samples. Because there's even other ICP-MS at my institute, but the problem is they measure sediment samples. And we are measuring carbonate calcium at the most pure level though that I could clean it. So we can't use a machine that it's being used for sediment samples or even water samples because it's not calibrated for the level of cleanliness that I need. So that's why it was so hard because we needed a machine that was basically only used for foraminifera or corals. Because even foraminifera and corals normally can't or shouldn't be used in the same machine at the same time because they need to be calibrated differently.
And all these allergies and so on, it needs to have good control and to
Exactly.
be sure because you cannot
Exactly.
go back, you are depending on that.
Yeah.
I totally understand.
Yeah.
I was thinking about the mycoplasma and PAAS, but it's the same. It's
Yeah.
used for a range of things.
Yeah.
Animal tissues as I use.
Yeah.
Oh,
All right, very well done. Our last presentation of this session will be by Jack O'Connell. Jack, whenever you're ready.
Okay. Hi everyone, I'm Jack, and I'll be presenting my thesis topic, in vitro gene activation in the fibroblasts of marine mammals. So, marine mammals include some of the largest and most charismatic animals on the earth. Unfortunately, they're also some of the most polluted, as apex predators and very long lifespans and high lipid content. They accumulate very high levels of pollutants, which, as you can see from this graph here, it shows some of the levels of persistent organic pollutants in marine mammals from Norwegian waters. See sperm whales, pilot whales, orcas, and polar bears are some of the most polluted. And we continue to release new and different chemicals into the environment with unknown effects. And we already know that some of these chemicals will have negative effects such as endocrine disruption, neurological and developmental disorders, cancer, and increased mortality. So it's really important as toxicologists and scientists to understand how these chemicals affect them. So traditionally, if you wanted to test specific chemicals, you'd use like in vivo testing or animal testing on model organisms like mice or zebrafish or mussels, and then you'd study the effects. But for marine mammals, due to practical, legal, and ethical reasons, it's not really possible. In general, though, scientists are trying to move away from animal testing and are increasingly adopting 3R principles: replacement, reduction, and refinement, meant to try and minimize the amount of animals being used in your experiments or remove them entirely. And just in June 2026, the EU published a roadmap to phase out animal testing for chemical safety assessments. So how are toxicologists going to get around this in the future? So it'll be used with a combination of in silico, so computer modeling, in vitro, working with cell cultures, and ex vivo, where you test with tissues taken outside of an organism. There are a number of benefits to these. They can allow much higher and throughput of different chemicals, much faster than you would from in vivo testing. However, there are still a lot of limitations with working with these models outside of the traditional, like, full animal. So it's really important to improve these toxicology models to identify new and emerging pollutants. Fibroblasts are increasingly being used as a cell model for marine animals. That's because they can be taken really easily from like skin biopsies with minimal impact on the animal. However, for toxicology, it's been found that or aryl hydrocarbons receptor genes can be repressed in mammalian fibroblasts. This is really an issue for identifying new pollutants, as the AHR pathway is really important as a detoxification pathway. AHR activates the transcription of cytochrome P450 enzymes, which are crucial for phase 1 biotransformation of xenobiotics. So this could potentially limit their use as a cell model. So how I try to get around this, I use triclostatin A, which is a histone deacetylation inhibitor. Histone deacetylation is a process that causes the chromatin to become more tightly binded. This can block the access for transcription factors like AHR from accessing the promoter region of genes. But TSA prevents this process, so the chromatin should be more open. This can allow the AHR to activate the transcription of genes like CYP1A. And so I wanted to investigate using this as a tool. So the aim of my thesis was to investigate using TSA as a tool to improve fibroblasts as a cell model to identify new AHR agonists in marine mammals using longfin pilot whales and polar bears. So my hypothesis were that pilot whales and polar bears will have low AHR gene expression to ligands alone, and that TSA will increase their sensitivity to ligands at lower concentrations. So I looked at two target genes, CYP1A1 and the AHR repressor gene, which sometimes can repress AHR genes by blocking AHR from accessing ARNT. And I looked at two known AHR ligands as a positive control, PCB126 and benzaprine. So this just shows how the skin biopsies were taken from Project Partners in Svalbard and on a voyage near Tromsø. Once the skin biopsies are taken, they can be cultured and used to make primary cells, which are refrozen and cryopreserved in liquid nitrogen. So I started off by expanding, unthawing these cell cultures and growing them more, and then I refroze them down to use in my exposure experiments. So just shows the pilot whale cells and the polar bears, and this just shows what I grew them in the T cell and pyroplasts. So to start off, I wanted to study what effect TSA would have on their viability. So I used the CFDA assay with resorcin, and I used different concentrations of TSA, and I also tested it with benzaprine just to make sure there's no combined effect on the cell viability. I then, for the main experiment, I took, for the viability, I just used PUA15, pilot whale cells. And for the main experiment, I used polar bear cells and pilot whale 9 cells, and I exposed them to 3 different doses of the ligands, with going like 10-fold lower each time. And then I performed qPCRs on them to measure their gene expression. And these shows the primers I was successfully able to verify. So for polar bear, I was able to verify the 2 target genes and also the 2 reference genes, GAPDH and ACTB, which I used to normalize the expression. And then for pilot whales, I was only able to verify CYP1A1 and GAPDH. So going on to the results for the cell viability, I found that higher levels of TSA can negatively impact cell viability, particularly for metabolic activity. And this effect was much more pronounced for when it was exposed for 24 hours or longer than, for 24 hours longer than the control where it was exposed to benzaprine and TSA at the same time. And it was also significant. And it crosses the 70% cytotoxicity threshold. So this just shows that it's kind of a threshold that's low enough that it's probably not just biological variation. So for future experiments, I pursue with the co-exposure, treating TSA and AHR ligands at the same time, because they have the best effects. So going on to the gene expression results, I found that TSA and AHR ligands did have significant interaction effects, with TSA leading to a significant increase in CYP1A induction with the AHR ligands. You can see even where it's not significant, it is much higher for AHR or CYP1A expression. And for like the BAP CH is much higher, even though it's not significant, and you can see it's across the board. And benzaprine leads to much higher expression of CYP1A than PCB126, which is just naturally how animals tend to respond, polar bears respond to them. Oh, I should say this is from the polar bears gene expression. I did find that TSA on its own leads to a significant increase in CYP1A expression. It's like 30 times higher compared to control, which is a lot. So it's just worth noting. I'll talk about it more in discussion. And then when I look at the ligands alone and ligands plus TSA, with for polar bears at least, they actually express CYP1A significantly to ligands alone. And you can see for PCB126, they're all significant. And they're all interesting at the same level, around 40 times higher compared to the control. And then for BAP, only BAP low wasn't significant and doesn't really seem to increase too much, but BAP medium and BAP high are significant, and they both increase interestingly and stepwise. But compared to ligands plus TSA, the fold change is much higher compared to without the ligands, and there is still significance between the two, but there isn't really a trend like in BAP. Like in benzoprine, there's no real trend with the concentrations. So I then, to try and compensate for the fact that TSA does lead to a significant increase in CYP1A, I went back and recalculated the fold change using TSA as a control instead of DMSO. And I did still find very similar trends. Like it is still, ligands do lead to a significant increase in CYP1A induction regardless. And then when I'm looking at AHR for polar bears, I found there was no interaction between TSA and AHR ligands, and only one treatment was significantly different to the control, and that was BAP high. And there's no real correlation between like BAP and PCB for the corresponding CYP1A induction, which is in line with previous research. And I think this might just be a random variation, it's a bit odd. For pilot whales, unfortunately, I couldn't quantify it due to the fact that when I ran the qPCRs, I had non-specific melt peaks and very high CQ values, which indicate there's probably really low target yield for CYP1A1. And then also for the reference gene GAPTH, I got like a very good qPCR results with like a specific melt peaks and like CQ values around 20. So there is good RNA yields. Thankfully, I was able to validate that CYP1A is being induced by pilot whale cells thanks to a Western plot. And this is quite limited because I only had was able to successfully do it with one exposure experiment, but it does seem to indicate that TSA does lead to an increased induction in pilot whales. Obviously, it's a bit limited. I normalize with actin, and there's a lot of background noise, but it does seem to indicate that. So, going on to the limitations. Obviously, pilot whales unfortunately could not be quantified, and I had a limited number of individuals used. I only had 2 pilot whale individuals and 1 polar bear, so that does limit the results a bit. And I had a very limited number of target genes, so it would have been nice to look at more AHR genes to see if they're also being affected differently or the same. And then obviously I had a very limited number of replicates, and some of them I had to discard due to, on the qPCR results, they showed non-specific binding. So this really limits the strength of statistical tests, so pretty much everything I show here should be seen almost like as preliminary exploratory experiment. And then there are some time and funding issues that led into that. So the time and funding limited the number of qPCRs I could run and the number of target genes I selected. And then finally, there wasn't enough time to go back and try and optimize the pilot whale exposures. I think if I tried and altered the seeding densities and the exposure time, I probably should have been able to get a proper qPCR run on it. And going on to the discussion, as exploring fibroblastic cell model. So TSA did lead for polar bears. TSA does lead to a significant increase in CYP1A induction by AHR ligands. And it did seem to increase the sensitivity to lower ligands with BAF low plus TSA leading to a significant induction, even when I recalculated the fold change with TSA as control. However, polar bears did show significant CYP1A induction by AHR ligands naturally, so I think it probably could be used as a model organism, cell model without TSA, which is probably better than altering the natural cellular processes. Pilot whales are much more complicated as I couldn't quantify them, and I had another qPCR for pilot whale 15 as a preliminary qPCR that had very similar results with non-specific binding and very high CQ values. When I went back and looked at both results, and I removed the treatments with non-specific peaks or incorrect melt temperatures, I found that it was only the treatments with TSA and ligands that remained. And then when I combined that with the results from my Western plot and also previous research project from the research group Emma Sud Jacobson in 2025, there is evidence that TSA could be used to increase AHR gene activation. And I do think it's worth exploring because another paper that used killer whale fibroblasts found that when they exposed it to different pot mixtures, CYP1A expression was actually downregulated, which is not what you'd expect and not what you'd want in trying to identify new emerging pollutants. So it's a question of why polar bears and pile whales responded so differently, with polar bears being very responsive and pile whales seeming to be epigenetically repressed, according to other literature. So it's possible that during cetacean evolution, they lost a significant number of genes involved in detoxification and the chemical defense zone. And polar bears haven't. Like, I think it's debatable if polar bears are really marine animals, but they have a much more robust chemical defense zone. And it's possible this could be an evolved defense against like pollutants, because chronic AHR activation has been linked to cancer and tumor formation. So this could be an evolved defense if they're being exposed to more pollutants and absorbing more. So going on to my conclusions. So polar bear fibroblasts are naturally suitable cell models for identifying AHR agonists without TSA. And from the results I got so far, it doesn't appear that AHR has any role in regulating CYP1A expression for polar bear fibroblasts, which is in line with other research from Pigs AL. They found similar results for human fibroblasts. And pile whale fibroblasts do seem to poorly regulate poorly express AHR genes, and this is likely caused by epigenetic repression from other literature I've read in my literature review and just previous results from other projects. So in conclusion, I do think TSA shows potential for improving fibroblasts as a cell model for toxicology, and it's worth exploring to try and help remove in vitro testing and further improve toxicology cell models. But it would require a lot more testing and optimization. So for future directions, like pile whales need a lot of optimization, and there can be a lot of biological variation between the two. I think there's additional techniques that might be more suitable, such as RNA sequencing and ELISA or luciferase reporter assay that might be better. And also methodology, try and use different cetaceans and add in new emerging pollutants. Thank you very much, and thank you to the Marma Detox and Environmental Toxicology team.
Perfect use of your time and a very dynamic presentation you did here. So I'm gonna give the.
No, it goes to me.
I go
No,
to
I have plenty
Patricia.
of questions.
Yes, I know you have.
So in this time it's really on my area because I'm cooperative endocrinology and I work in endocrine disruption. I know how difficult it is to set up new. This is a non-new approach in methodology to replace aminos and so on. And I have some doubts, and I know all this takes time to make new cell lines, isolating, and so this will be based on isolating the fibroblasts from the polar bear biopsies, skin biopsies, and it will be primary cell cultures isolated and used at each time, isn't that?
Yeah, so these samples were taken actually in 2024 by project partners in Svalbard for the polar bears and then up near Tromsø for the pile whales. So they were already cryopreserved, and like pile whales were done by Sophie Soderstrom, who was a postdoc in Marma Detox project. And so they were already isolated and cryopreserved before I.
Oh, that was my question. So quite stable and so on. Yeah. Because we were testing in fish pituitaries or skin and so on, but taking it each time. So this is good, so you can preserve the sample and so on. Then the next level is you test the disruption of the HRR signaling based on the CYP1A1 expression, isn't that? So it's not based on receptor assays or anything or reported gene. No, it's based on the gene expression.
Yeah.
Okay. And which means that if you want to take for other species, you would have to optimize the qPCR species by species, isn't that?
Yeah, and I've also found that this
For
even
comparative
between individuals it can be very different. So between like the PWAL15, I did another qPCR, and even just growing them they grew very differently. PWAL15 grew really quickly, and they proliferated to 90% very quickly. But PWAL9 took way longer, so it took much longer to grow. And polar bears grew way differently as well. So even between species and between individuals, there's a lot of variation. So I think that might be why my results, I couldn't quantify my qPCR compared to Emma last year. I think it was just we used different pilot whale individuals.
And then it takes a lot of time to identify other markers that could be by enzymatic expression, enzymatic activity, and so on. I have that experience with non-invasives taking the fish scales for endocrine disruption, but then a lot of variation and so on. So in terms of the cell's activity, then the TSA affected cell variability, which is not a very good thing. How did you measure that? It was metabolic activity, you said?
Yeah, so I used the resorcin CFDA-AM assay. So resorcin is like a proxy from metabolic activity, and then CFDA is like membrane stability. So.
And what is the solution? So I think you said that you could use TSA as a control instead of DMSO, but it is also diluted in DMSO, the TSA, the compound?
Yes, dissolved in DMSO. So for my control
so
cells.
So you have the control that would have both things?
Yeah.
Growing, okay. And by curiosity, did the DMSO affect the metabolic activities of these cells? No.
Well, I used it as the control, I use the DMSO as like a reference point.
As a reference, yes, but sometimes we do it without the DMSO, and now we have some stable cell lines with the estrogen receptors from fish, and the DMSO itself is affecting. You did not test a control without the DMSO?
Oh, no.
No.
Yeah, I didn't just use cells.
That could be an idea, because sometimes even the DMSO is affecting depending on the site.
Yeah.
Let's see, qPCR was with stable reference.
Maybe the last one.
Last one, otherwise then I can ask later. And basically maybe I'm taking the question from somebody, what was the doses? They were based on environmental levels in bibliography or measured in the group?
Yeah. Oh, so the environmental levels, they were just like.
You just say medium, high.
Yeah. So where did I put it? Yeah. So honestly, these were just kind of like general levels used by the department. So a lot of the time they use this, and I just lowered it tenfold each time to try and see if there was like a stepwise increase or if there's concentration effect to the expression.
But they were based in incremental levels or accumulation levels?
Honestly, it was just things my supervisor said.
First trial,
Yeah.
when the beginning.
Yeah.
Okay, and then.
Thank you, Patricia. Who wants to go next?
I can go. Okay. So congratulations for such a nice work. I mean, it's a lot of data. It's really hard to present in 15 minutes. So I think you did your best. So well done on that. So one of my questions was related with the doses. So where does concentration and how realistic those concentrations are? Because you could have really clear molecular response, but in unrealistic scenario. So the idea is to try to adjust to a realistic scenario. That's why it was. I can agree with that. The other thing is, so you say, I mean, you have to candidate. Of course, it's somewhere you have to start from somewhere. But have you considered instead of a candidate approach, go more to transcriptomic approach? Or
Yeah. So part of the reason I didn't is because like funding issues. So I think RNA sequencing might be a better solution than qPCRs, but I'll use phrase reporter say, but just for funding and like, yeah, I wasn't able to do it, and there wasn't the opportunity to do it in the lab.
yeah, also because it could be nice to combine both epigenomics approach and transcriptomic to see how the effect of, because you are suggesting some epigenomic regulation, so how clearly, if it's a clear association between some epigenomic changes or differential methylation regions with the expression of some, and also the idea, I mean, it's all, I mean, every study has certain limitations, so you have to start from somewhere, and in that sense it's great, but when you have different genes that maybe even are in the same pathway, so you can really test if that is affected or not. But I think this is a starting point. So in that sense, it's great. And the other thing is, out of your opinion, how different is a rat or a mouse in terms of response with these birds?
With this, with like CSA.
Yeah. So how different are they?
They can.
What are you what are you gaining by doing in a polar bear culture than in mouse or rat?
Yeah. So for at least for AHR ligands, like sometimes they don't. It can be different depending on the species. So it could be good if you just want to be like, oh, it's toxic. But I read another paper about like comparing AHR ligands for polar bears and humans were much more responsive, and like a lot of the ligands they tested that triggered our AHR response didn't activate in polar bears. And I do think there could be some benefits if you're trying to regulate chemicals to have like, oh, does this to killer whales or does this to belugas, compared to mice because they're very charismatic species and people tend to care more. So if you are trying to get a bit of a movement going to regulate them, it might be better to be like, it does this to this species.
Because I would think it's nice that if you can show that.
Yeah.
So if you have the comparison to see, that's why we need to do this, because the response is different. It's a way, if you compare with something with mouse, rat, that is cell culture, even the same type of cells, of course, to see that's why we need that. It's a way of justifying why all this work that you did is necessary. So in that sense, it's great.
Yeah.
Thanks a lot. Great job. Well done.
Thank you.
Thank you, Gonzalo. Which one of the Negroes want to go first? Don't fight.
Yeah, up to you.
Okay, thank you.
Really good.
Thank you. Well, Jack, congratulations for the presentation. I really enjoyed the way you presented your research. I have a couple of questions. The first one is that if you ever thought in playing with temperature here, because this stressor, contamination, pollution in general, doesn't come alone, and especially in the Arctic, it comes with a lot of temperature increase.
Yeah.
So, would you do that, or if you have some money for doing that, of course, and time? And what would be the results that you would expect?
I don't think I'd alter the temperature because, like, obviously marine mammals are warm-blooded creatures and, like, they've got a lot of fat inside them. So when we were culturing the cells, we kept them incubated at 37 degrees, which is their natural body temperature. I don't necessarily think that once the chemicals are inside the body, they'd be, like, at a different temperature. Maybe I don't know what the skin temperature is versus, like, the internal, like, organ temperature is, but normally, like, they keep the fibroblasts growing at 37 degrees incubation.
My other question is related to this defense zone you mentioned. That the immune system of the pilot whales should be probably weaker than the one from the polar bears.
Yes.
Why is that, and why is it related, or why do you think it's related to the evolution of marine life?
Yeah. Well, I'm not really sure because this is like pre-published research by like another person connected to the Marma Detox project. They were like looking at all the genes of marine mammals, and they found that cetaceans have lost like a significant number of genes completely, and a lot of the remaining genes are pseudogenes, so they're like inactive. And I don't really know entirely why, but just thinking about it myself, I think like they have a lot of fat, so even before humans, there would have been more chemicals in the ocean, they would have accumulated a lot more. So it might be that they did lose it just because if they were constantly responding to all these chemicals they're absorbing in the water and eating in the fish and accumulating in their fats, they might have negative effects because AHR activation is linked to cancer, and especially if like they're constantly having, and especially chemicals like PCB126, they don't get metabolized, they just stay in the body and keep activating AHR, which I think why is like Ben's-Priem response and that response is different. So I think it might be just an evolved defense. Like if they did have this very robust defense system, they might just constantly be going on nonstop. And like even for fish that like live in very polluted waters, they tend to have their AHR pathways repressed.
Okay, thank you very much.
Thank you, Nico. And so, Nico.
Thank you. I guess I'm gonna be quick because I'm way out of my depth with precision is absolutely not my field. I must say I don't know to which extent it was due to me being absolutely not knowing anything, but I was quite quickly lost with the presentation about like what was the, in terms of the graphic presented, what are the response variable, which is treatment, etc., etc. And I think perhaps that could have been better explained, at least for me. I was totally lost. So I guess I got a very stupid ecologist question for you. Are you sure you work with pilot whales?
Yeah.
because I think that in your photo in the introduction it's a sperm whale.
Oh yeah, this is like probably, but this is just like from the cruise. I don't like.
It's
just
I'm sure.
Like this is just
No worries.
this just shows how they're taken for the whales.
But I think those are scum.
Yeah, probably. But this just shows how they're taken. They just
Yep.
go up on, they just ride by and shoot them with a dart and
Yeah.
take the skin. It's not the most nice way to do it, but I don't think being knocked out and sedated is not very nice either. But yeah, this is just how they take it for the whales.
I guess that's it.
So, Patricia, if you have one or two lingering questions, quickly. Maybe one for, but go ahead, Manuel.
There's one. What about the molecular? Because somehow you're measuring molecular response, yes? And the long-term molecular response is changing after. Because what you have in the system is you expect some bioaccumulation, so
Yeah.
the level increase. So what happens with the long-term response when you remove or you increase? So have you tested that somehow or not really, or it's just after exposition and then you know what I mean?
Are you talking about exposure to TSA or the ligands?
Or even both.
Yeah. I tried to do like a kind of pre-exposure where I just like gave it TSA for 4 hours and then remove it. But I tried to validate with a Western blot and it didn't work. And at least for this, normally when I was finished with them, I just lysed everything. I just RNA lysed them all for the qPCR, so I don't have anything afterwards.
Sometimes it's nice even to keep part of that if it's feasible, and then to analyze like if it's how plastic is with the response. That's my question. Yeah.
Yeah, I'm not sure how I keep half of it. But yeah, that'd be worth exploring. But I know that at least keeping half of the fibroblast might be difficult because I found that like part of the reason I used 2 piled individuals is because when I was growing the first one, P19, they ended up turning senescent when I tried to grow more cells, so I had to use different individuals. So I think if you just left half of them, they probably wouldn't keep growing because these fibroblasts, they're not like immortal. Eventually, they will just stop growing and lose their morphology. So I don't know if that is practical for this thing.
Right. If there are remaining questions, I will have to wait for the coffee break, Patricia. So congratulations.
That's fine.
Congratulations to everyone in this session and keep up and congratulations to the ones that are going to come to the last session because they are the most resilient.
You tell me when to pitch.
Yes.
We're
Live.
We're live, okay. So welcome back to this final session. My name is Gauthier Chal, I'm from UBO in Brest, so I will be chairing this session. Let me first introduce the jury. So we have Nico Weinberg from the University of the Asturias, Oviedo, Gonzalo Machado from the same university. We have Iker Martinez from the local university of Basque Country, and Nico Jegre from the University of Brest in France. So Kate, you have 15 minutes to talk. I see you have a timer, but I will still wave at you when you have 2 minutes left so that you know it's time to conclude. So the floor is yours.
Thank you. Hi, everyone. Welcome. My name is Kate Logan. I'm presenting my thesis from Consumption to Coastline, assessing the ecotoxicological effects of the antidepressant sertraline in Mytilus galloproventialis. I wanted to start, I was supervised by Stefano Gorbi, and this work was performed at the University Politecnica della Marche at the Lab of Ecotoxicology and Environmental Chemistry. To start, I want to give a brief overview about pharmaceuticals in the environment. So they're currently considered contaminants of emerging concern. This means that they're understudied or unmonitored, but they're suspected to pose a risk to marine environments or to aquatic biota. I've included a little graphic here that we can follow how when pharmaceuticals are typically not fully metabolized by the human system. So many times when you take medication, it excretes directly into the wastewater treatment plant in large shares unchanged, and its metabolites flow to the wastewater treatment plant, which is not currently equipped to process or to remove them from the wastewater. As a result, there's a direct discharge of high concentrations of these pharmaceuticals into surface waters, rivers, and estuaries. Of the pharmaceuticals that are detected, antidepressants are quite prominently represented. In fact, this is following increased consumption and prescription trends in the past 2 decades, with a spike noted after the COVID-19 pandemic also. As you can see here, it's just a good little pathway. And of course, moving into a more particular antidepressant, I'm going to be talking about sertraline, which is an SSRI, or selective serotonin reuptake inhibitor. It's commonly used to treat depression and anxiety, and it's one of the most commonly prescribed of its class currently in the US and in North America. So sertraline works by, as the name would suggest, blocking the reuptake transport of serotonin, resulting in a higher concentration in the presynaptic membrane. This is therapeutic for humans, for mental health conditions. However, the serotonin pathway is conserved across taxa, so many invertebrates still use serotonin for different neurotransmission and physiological kind of reasons. So that SSRIs can remain biologically active in marine species, and existing literature suggests that sertraline itself can induce behavioral changes, oxidative stress, developmental alteration, etc. It's been particularly observed mostly in fish as well as crabs, but I'm going to be focusing on bivalves, which have not been as extensively researched. So we devised the research question, can sertraline induce biological effects in the Mediterranean mussel, Mytilus galloproventialis? We chose the Mediterranean mussel because it's widely geographically distributed. It has been documented in a variety of ecotoxicological assays already, and it's a model organism for studying the kind of accumulation and the responses to contaminants across a broad scheme of typologies. So our experimental plan went something like this. We studied 4 concentrations of sertraline, 3 kind of representing our environmentally relevant concentrations. The European Pharmacy Project has found detections of up to 200 nanograms per liter in surface waters in the Mediterranean area, which is a moderate level for this kind of drug. And we added the 500 nanograms per liter as a dose-dependent testing kind of thing to to kind of contextualize the environmentally relevant doses. We performed a trial of 14 days of exposure to sertraline. This was preceded by a 5-day depuration period, and then digested the mussels for digestive gland, hemolymph, and gill tissue. I tested a total of 19 biochemical and cellular biomarker assays, so I'm not going to go into all of them today, but I have listed them all here for you if you're interested. But they encompass 5 main systems: the immune system, genotoxicity, neurotoxicity, lipid metabolism and peroxidation products, and antioxidant defenses. I've just put a couple of photos here from throughout the process, and I'll get into the results. So, for the immune system, we studied neutral red retention time, granulocyte to lymphocyte ratio, and phagocytosis capacity. Neutral red retention time is a measurement of lysosomal membrane stability. So the lysosome is obviously a main target of these drugs. As you can see, it was significantly compromised at the 200 and 500 nanogram per liter level. So this has been observed in other antidepressants, also in the SSRI class. It's suspected that this is lysometropism at work, so there's the accumulation of these drugs in the lysosome, and this weakens and destabilizes the membrane, which can have a cascading effect on the immune system of the muscle. In tandem with that decrease, we saw an increase in the ratio of granulocytes to helinocytes. Granulocytes and helinocytes are two types of hemocytes that exist in the circulating in the hemolymph of the muscle. Granulocytes are responsible for phagocytosis, which you can see also, though not significant, increased along with the granulocyte proportion. So just to validate that. And the helinocytes are associated more with an encapsulating like coagulation response. So this can indicate that the muscles are having a kind of inflammatory response to the sertraline in the system. On to genotoxicity. This was a little bit less exciting, perhaps. We saw that there wasn't any significant results here, but you can see there's a little bit of an increase when you get to the higher levels, 200 and 500 of sertraline. However, there's a lot of variation, and these also were below the kind of natural seasonal variability of the muscles. Usually about 1 per 1,000 cells for micronuclei is relatively normal, and up to 40% of the cometus A is pretty normal. It's relatively more reversible, versus the micronuclei is a bit more permanent, like existing damage. Next compartment is for neurotoxicity. We tested the activity of acetylcholinesterase in both the gills and the hemolymph. Hemolymph proved to be less sensitive than the gills, and the gills showed a bit of a non-monotonic response, you can see here. So the 0.2 and the 500 nanograms per liter were both depressed as opposed to the 20 and 200, as well as the control. Acetylcholinesterase is an enzyme that hydrolyzes the neurotransmitter acetylcholine. And acetylcholine is responsible for a variety of different functions in bivalves, ranging from different muscular contractions, ciliary beating, heartbeat, and catch muscle regulation, which, remember that, because we'll come back to that later. And moving on to lipid metabolism, we studied the lipofuscin content and the neutral lipid content. So these are two pretty classical biomarkers for lipid peroxidation products. So lipofuscin has more of a historical wear and tear kind of connotation, and this is a bit more permanent damage. So as you can see, at 0.2 and 500, this was depressed, and at 20 and 200, there seemed to be more accumulation of the lipofuscin in the digestive gland of the muscle. Neutral lipids content followed a very similar pattern, which was surprising to us, but it reflects kind of a more current disruption to the muscle's metabolism. So this is a collection of unsaturated lipids in the digestive gland that, on their own might not be super harmful, but in greater levels can become kind of inconvenient, one could say. And once again, following that kind of trend here. This could be used to interpret like a breakdown in glycogen utilization or in redistributing existing lipids. The final compartment I'm going to talk about today is the antioxidant system. This one is very nuanced. It's a bit difficult to interpret for ecotoxicologists. We tested 5 single antioxidant enzymes, and of those 5, glutathione S-transferase and glutathione peroxidase were the 2 with significant results. At the 500 nanograms per liter, you can see that they were significantly depressed, although interestingly, in comparison to the 0.2 nanograms per liter. And both of these utilize unconjugated glutathione to kind of remove things that might be more harmful or interrupt like the processes of the muscle. So like I said, these individual enzymes are quite variable, so we utilize total oxyradical scavenging capacity to kind of add a level of organization. So this measures the ability of the muscle to kind of defend itself from reactive oxygen species production. And as you can see, this was also depressed from the control at the 500 and the 20 nanograms per liter. So this indicates overall that the muscle is coping with the stress that sertraline is inducing. However, the fact that it is depressed
And as we know, contaminants are rarely solo players. It could be a kind of difficult situation for the mussels if there are more, obviously, climate change, ocean acidification, more contaminants. It could be very difficult for them to cope with the stress. So, looping back to the neurotransmission, because I believe this is part of the core of the issue and trickles into the other parameters that we measured. As we know, serotonin is increased when there's an SSRI in the system. There has been a pretty well-documented relationship between the serotonergic and the cholinergic systems. So it's been shown that in higher concentrations of serotonin, the acetylcholinesterase enzyme becomes a bit less active, at least in bivalves. And one could posit that there is a increase then in acetylcholine availability. I'd like to preface that we did not directly and explicitly measure these molecules because it's a little bit more complex and limited time, all that fun stuff. However, this could induce several changes in physiology. In particular, one that I thought was interesting and could warrant some future study is in the calf muscle. So when mussels are able to force their shells closed and kind of lock their muscles in place in a way that is useful for protection from predators, protecting from desiccation, and it uses very little energy expenditure as opposed to an actively contracting muscle. So it can range from periods of minutes to hours in life, and they go in and out probably several times a day depending on the conditions. So the thing is, acetylcholinesterase removal is what initiates the catch state, and the kind of elevated serotonin levels are what relaxes the catch state. So if both of these neurotransmitters are present in altered levels, it could have some implications for future access to that catch state. So if it's a longer period of exposure to sertraline or other SSRIs, there could be some effects for being able to access this very important state for energy. It could change energy expenditure, it could change metabolism, the production of reactive oxygen species, it could increase stress. There's a lot of things that could be greatly influenced by the changes in these molecules. Now, one does have to wonder, where does this all kind of come in? They're just muscles, they're pretty small. We determined that sertraline exposure has an observable effect on the immune system, cholinergic system, and metabolism in Nucleus galloproventialis. But in the bigger picture, SSRIs and other CECs are already quite widespread. This graphic here is kind of showing average maximum detections of different antidepressants across the world. And as you can see, in Europe and North America, it's a pretty moderate, to moderate high level of these available, and you'll notice that there's several of them in each of these places. It's not very often that you see there's only one drug present. So as you can see, there's several even within the same typology of drug. So this shows us that they're co-occurring in the environment, and there's an interesting thing that individual contaminant studies like the one we did are able to aid us with future analyses of these contaminants that we don't know very much about. So for example, my work was helped a lot by analysis of fluoxetine, which was just added to the contaminant watch list for the Water Framework Directive this last year. So it was a concern for their chronic toxicity, and this led it to be kind of designated as a target for future water monitoring programs. So in short, we do need more future study into chronic exposures, because the reality is also that these drugs are being recharged constantly based on the wastewater treatment plant output into surface waters. And not only are they no longer really emerging, but they are here to stay. Thank you for listening to my presentation, and I'll open up for any questions.
Thank you very much, Kate. Who wants to start? Nico.
I can start, yes. Well, thank you very much for the presentation. It was really nice. I like the way you addressed the public and presented very, very nice. It's true that I missed that some of the figures could have been bigger. For example, your last map, it was very concerning.
Yeah.
This should take the whole screen.
Yeah.
Because And talking about this map, which is the area of the world that you are concerned most? Not
I personally, or that
in
project?
general,
Pretty bad.
not only because the present map, but because of the future.
Mhm.
How is this going to evolve? Because it's not going to evolve probably homogeneously
No,
all around the world.
not at all.
So,
No, that has a lot to do also with the treatment of wastewater. So currently in the EU, I think it's something like 80% of households in like areas of over 1,000 inhabitants have secondary wastewater like treatment, and it is mandated by the EU that you have to have up to secondary treatment. But it's implementing that is difficult. So especially in more rural areas with less people, like that can be a change. Also, the fact of the matter is they're trying to move towards like tertiary treatments to remove nutrients, but even tertiary treatments are very difficult. Like they don't exactly address all of these pharmaceuticals are very complex. They all have different properties, so it's very hard to remove them from wastewater with like one method. So multiple methods, probably more like cutting edge, newer. It's very expensive, and also if you're handling a lot of wastewater, it's kind of difficult to filter that much in that amount of time. So I think it's going to change for the worse probably, but it's also like seeing how the consumption trends are going. I think we're only taking really more medication. I doubt there's going to be a change to that. So the fact that the wastewater treatment plants are not going to be able to adjust very quickly is also kind of compounded by the fact that we're still taking a lot of medication, and neither of those are going to change very soon. Yeah.
I have a question on your stats. Can you go back to your bar graphs?
Yes.
This one, yeah. So here you are representing the standard error or the standard deviation?
Standard deviation.
The standard deviation. So you have.
I'm sorry, I should have mentioned I did one way ANOVAs for each of these.
Yes, but probably you don't have homogeneity of variances, right?
I did. I tested. I mean, it's
It
kind of
looks-
weird, but
It looks like there is a lot of variation in this
There
bar,
is.
no?
yeah. I
And
mean,
you have in all of them.
yes. The thing is, the samples are kind of difficult because I had a total of 60 mussels in each tank,
Mhm.
and each sample was 3 individuals. So they were kind of pooled together. And then for each test, I had 10 samples. So as a result, we're looking at kind of like 30 mussels. And each sample had a lot. There could be variation from individual to individual. So especially for the genotoxicity biomarkers, some of this was also related to slide preparation. I think I had a little bit of difficulty with getting the method down for this one. It was I had to learn all of these protocols from scratch. So I think probably as you can see, there was the most variation because often this was counting like 1,000 cells, and it was basically like 2 micronuclei, 1 micronuclei, 3 micronuclei. So the counts are also quite small. I think that is kind of a difficult thing with this. So there's a lot of individual variation, but there's also probably some like variability in like the sample preparation. Yeah.
Okay, thank you very much.
Thank you very much.
Here we go. Gonzalo?
Right. Yeah, thanks a lot for the talk. I really enjoyed it. So well done. It's many different experiments, so it's a lot of work. So well done.
Yeah.
On that. So and now you're saying that you did most of that, though. So
I did all of them.
So that's
Sorry.
a lot of work.
Yes.
Congratulations on that. So I have a few questions. One, you already answered to me that it was 60 muscle per treatment, so a group of 3. So that's great. Have you considered, since you have the individuals and you can even make it individualize the samples,
Yes.
to measure some sort of, because you talk about response and how some condition factor or something like that. Have you addressed that?
Yeah,
Because you didn't show that part, I guess. No?
yes, you mean like the kind of seasonal conditions or the like?
Well, how after the treatment, how they change in terms of I don't know, some weight,
Yeah,
some
we
condition factor, typical condition factor, or something like that. Yeah.
we didn't do any kind of like physical measurements aside from shell size, but that like wouldn't have changed in 14 days.
Yeah.
I mean, it also was quite a short exposure period, so I think the most like easily observable would have probably been the 14. We dosed the sertraline every 2 days and then fed them every other day, so between the dosing, so that we would try and minimize the kind of like water mixture being too, like either running out of chemical or running out of food was a little bit of a balancing act. But yeah, I think it was a little bit difficult to measure physical parameters. Although we did measure mortality. I think anecdotally we maybe lost 5 muscles in the process. So, but I don't think those were entirely sertraline related.
And one question is also related with the methodology.
Yeah.
The way that, particularly for the genotoxicity,
Yes.
the way that you address was completely random. So you just, without knowing, completely blind, so you don't know from which treatment was that you were analyzing, or you always have a track.
I did know which treatment it was from, so that was perhaps a bias. I did also have my supervisor, she took, randomly chose a slide from each treatment level and like spot checked me to see if like the values were relatively high. So there was like a level, but my count data, I did know which samples were which ones.
Okay, and just this is more a suggestion than a question. So,
Yeah.
because in your case, it seems that at least for this particular sertraline,
Yeah.
it's the concentration is quite realistic. No,
Mhm.
correct me if I'm wrong, but there's some places like in Canada or something like that.
Yeah,
it's
they're quite
410,
high.
no, correct me if
Yes.
I'm wrong, and it's the same unit, yes.
Yes.
Nanomolar per megagram. So I want to even start with that to say it's important to test realistic concentration, and what
Yes.
you did was quite realistic. So it's already, and we know that the situation is going to be worse in the near future. So in that case, I think it's quite nice that you adjust to real concentration. So that's it. And of course, if you have in the future some opportunity to go like more long-term exposition, I think
Absolutely.
you could have maybe this significance that are suggested somehow. So
Totally, yeah.
so great job. So thank you very much.
Thank you. And if I could just briefly speak to that, they actually are doing a follow-up experiment now with higher concentrations, in fact, like into the microgram per liter level. So you're trying to test. But the reason I chose these ones was because that was the local area, because it's obviously not a static value. It changes a lot. So this was in the Adriatic Sea, like near Ancona. So it was kind of locally devised. Like you're not going to find 500 in Ancona.
Yeah.
Yet. But
Not yet.
Yeah.
Thank you very much.
Thank you, Gonzalo. Yeah.
Thank you, very interesting. And so a few questions. For the method,
Yes.
how did you keep the muscles? Was the water recirculating or?
They were each in their own individual tanks. It's a little bit difficult to see, but I have bubblers in each of them, and we had to do water changes after each. So we dose, then we do water change, and then we feed. So that was kind of the schedule. Was Monday, Wednesday, Friday was dosing with sertraline, and then Tuesday, Thursday was doing the water change, and then the
So you were changing the water and adding.
Yes.
Yeah.
and then recirculating with the bubbler. So that was they were all closed systems. They were not flowing through.
Another question. There are, as far as I understand, 2 types of these serotonin uptake inhibitors. One of them is used to treat depression and anxiety, so it kind of calms you down.
Yeah.
The other one is to treat only the depression, so it gives you a high.
Yes.
Which one is this?
This one is a SSRI, so it's for both. It's a combined therapy. I think that it's typically prescribed. It can be prescribed for anxiety, depression, and a variety of like in that category of drugs.
The one
There's
that calms you down.
yes, yes. So to treat like shortages of serotonin, like supply naturally in the brain. So it's more of a to help you like produce more calming like effect. Yes.
Maybe the other one you have a different.
This is also true. Yes.
Yeah, and then the other day also usually they are, you don't get one of these.
Yeah,
drugs in
I
the
know,
bottle,
yeah.
you get a lot of them. Then, did you see, because 14 days is probably also a short time. I would also, next time that you measure, you should not measure the size of the shell, because in 14 days the shell will not drop. It's the animal itself, the one
Yes.
that can be affected.
Yes.
Maybe they are getting less or whatever. And then another question, which is, back to the figures, the statistics, and the huge standard deviations and all that. Maybe it would be more visual if you present, instead of the average, the median, the box plot, because then you can see if the points are up or down, or if they are, so then it is, because some of these, even if they are not significant, there is a very clear trend.
Yes.
So maybe with more individuals.
I initially did want to use box plots, but the way that, like talking to my supervisor, this is the way that they've kind of got buttoned down to do it. So there was a little bit of a collaborative effort there where I think this is the way that they think with, it's more of like a diagnostic, like you can see kind of, it was a style thing, I think, also for them. So, thank you.
Thank you. I like the presentation. It was very clear. One thing that I really liked, and I think that's not necessarily many people do it, is that you've got a lot of different things that you measured, but that I know nothing about. But you managed to group them in very clear blocks and groups, so that's good. That really helps understanding. So thank you for that.
Mm-hmm.
Related to that comment and still on the statistics, so you did one-way ANOVAs for each factor. Do you see a potential issue with that? Statistically speaking.
Statistically speaking, I mean, there's a couple, but like I for this, I kind of stand by it, I think.
Okay.
In this case.
I think it's fine for your result because you've got a pretty clear pattern, especially in like the one before. There's like clear those effects and all of that. One risk you have when you do your statistical analysis this way is that you do multiple testing.
Mm-hmm.
And so with a standard statistical test, you've got basically one out of 20 chance to say, oh, there is something when there is nothing. And so if you test a lot of things, you are bound to find something significant.
That's true.
But I think that's all right because you've got clear feelings now. So
it is a bit nuanced. I mean, especially with the antioxidant one, where I think there is a lot of variation. I included my graphs at the end where you can see kind of wrong way. You can see kind of like there's quite a bit of variation in terms of the enzymes were all over the place. I feel like that it's a little. You can see that there also was not like significant stuff going on. So I did test quite a few. Like I think I showed 9 graphs and I did 19. So I think there were a good number of tests that also just didn't really show anything. So these also I did try to validate also with literature and at least for like other SSRIs this seems to be relatively.
No, again, I don't think this is a big problem for you. It's just like
I appreciate it. Yes, thank you.
The general practice, I guess. And then I've got a much I don't know if I have too much time, but
5 minutes.
Wider. Oh, that's all right.
4 minutes. 4 minutes.
A much wider question then.
No, sorry, Kate.
That's okay. I know
I should have lied.
No, but much wider question. So with this experiment, you measured just the endpoint after 14 days.
Yes.
And I was wondering, would you expect the muscle to possibly adapt and correct the response if you got more time for them? Do you have any idea about that?
Yeah, so actually there is some interesting, I would say there's some interesting kind of implications here with the curve that we're seeing because you can see at the 500 nanograms per liter, there's like my theory is that, and what has been backed up a bit, is that there is the potential that they might be able to modulate to the exposure to an antidepressant. However, this would take a lot longer than 14 days. I mean, I think that obviously you see muscles growing in very adverse conditions. Like it's possible that they could become more like able to kind of defend against the negative impacts. I think also in particular it'd be interesting to kind of study the ones that are closer to hospital outputs because they have the highest by far, like in terms of export of like the discharged water with concentrations. There's like, I think maybe microgram level of sertraline in those. So I think it'd be interesting to see muscles that are able to like survive in that long of an environment, if they exist, their distribution, how common that is. I think that it's possible that they could, but there's also a bit of a problem with the lower doses because at the higher doses, it's almost like the muscle is able to sort of adapt and change its, like, there's a possibility to kind of modulate its serotonin production in response to the kind of continued suppression of its reuptake. However, at a lower dose that might be kind of more inconsistent, it could be difficult because they're not constantly exposed to that higher level. So that's why acute exposure is also kind of an interesting thing to study because it could be that the water's circulating and it's just like for a few days it's really like rich in sertraline and then it's gone. I don't know. I think the bottom line is that I think longer time periods would be very interesting to see how this would work with sertraline and also with other drugs. So.
Thank you, Nico. Congratulations, Kate.
Thank you.
And then we continue with the last two presentations on birds. The first one being given by Emil. So you know the rule? At 30 minutes, I show you
the peace sign.
the victory sign.
Fantastic.
Whenever you want, Emil, the
Yes.
floor is yours.
Thank you very much. And hello, everybody. What a pleasure to see you all. I have now also the pleasure to build a bridge between mussels and birds, as I have a little bit of both. My name is Emil Eichenbrunner, and my topic was the habitat use variation of the common redshank and the effect of shellfish exploitation. Let me start by giving you some context of why it matters to study habitat use variation. That would be that wetlands are being lost globally on a very big scale, and these losses are also found on populations of shorebirds that depend on such wetlands. Furthermore, wetlands are very complex habitats. Not only are they in fact a habitat mosaic, so various habitats in close vicinity to each other, but also each of these habitats changes across cycles. Environmental cycles, such as the tides, that can determine the availability of, for example, the intertidal areas, or seasonal cycles that will determine or change the profitability of certain habitats. And considering all of this, if we want to reverse the trends of wetland loss or shorebird population losses, we will have to consider habitat use variation, and conservation must include this knowledge into conservation efforts. Now, I studied the common redshank. It's a flexible wetland generalist, which means this medium-sized wader can forage on a huge variety of prey items, so different prey in different habitats across cycles. And they are sadly they're having population declines across Europe, but especially in Portugal, they have local declines. And what you can see here is the migratory route between the breeding seasons and the non-breeding seasons. So similarly to us university students, they travel across Europe to connect different places in their life cycle, and they will spend their winters in Portugal, as I did. For example, in the Thesis estuary and the Ria Formosa, which are two of the most important wetlands in Portugal for waders such as the common redshank. They are both protected under various national and international levels due to their importance for different habitats, but also for different communities of different species, especially waders. And what is also important, especially for my study, is not only the variety in wetlands, but also the presence of shellfish exploitation activity. So two different types of shellfishing that occurs in both, and that would be the shellfish harvesting in Thesis estuary. So shellfish harvesters that go through the mud in a mobile way to collect shellfish. And in the Ria Formosa, we have a more fixed and long-term and stable environment of the shellfish exploitation, which would be aquaculture plots. So we have fixed structures in about 40% of the area of the Ria Formosa, where they culture shellfish, while the shellfish harvesters of high densities, they cover 13% of the Thesis estuary. So quite important areas, and I'm only here reflecting the high concentration of shellfishes. The numbers have increased dramatically the last 15 years, so 70 times fold is nowadays the activity. But more about this later on. Now, all these GPS locations are the ones that I was able to use for my thesis. They were obtained by GPS devices, so little devices that were attached in a sort of a backpack to the common red chains, 19 individuals in my case, that were of both regions combined. And they sent locations during 6 non-breeding seasons, so the last consecutive 6 years, and gave me a total of 162,000 GPS locations that they had the pleasure to analyze. And this is the nice approach of GPS satellite tracking that allows to answer ecological questions across regions with reduced effort. And how exactly did I go on? What were exactly my ecological questions? Well, first of all, concerning habitat use, I tried to answer what is the variation in habitat use across regions, day, night, the seasonal periods, and also tides. Concerning shellfish disturbance, I wanted, and I did answer if redshanks used or avoided areas of shellfish exploitation. And I went on to answer these questions by first receiving this gigantic data set, cleaning it digitally, so getting rid of locations that were, for example, technical errors or outside of my study location. I annotated them with all information of interest, such as being from a certain tidal stage or during day or night. I further on overlaid them with habitat and shellfish exploitation maps, which allowed me to again categorize them as being from within or outside of a certain habitat, or like what type of habitat or what type of shellfish exploitation activity. And once this was done, I had an even bigger data set that I used to calculate the proportions of habitat use. So I started with individual proportions. So a percentage that would tell you, for example, how much did this individual use intertidal flats during low tide, and so on, for each habitat across all categories of all cycles. Once this was done, I went to my favorite part, not the statistics. I did linear mixed effect models to test for significant differences between those proportions of use, to see which factor had how much of an influence on the habitat use. And the EVLAB activity index was used to assess whether they indeed use the areas or not. So that's an index that from -1 to +1 that will give you a degree of preference for these areas in relation to the availability. So that was a lot of statistics, lots of coding, I would say way too much, but it was worth it, as I had very interesting results, starting with, first I'll explain you a little bit of the plot. We have here the habitat use proportions, percentages actually, so each bar represents one habitat with its column. Tagus estuary on the top, Ria Formosa on the bottom, and categorized into different categories of each cycle. In this case, a tidal cycle, with an overall overview of the habitat use for both regions. You will see that we have a huge variety in habitats simply. There's a different availability between habitats, so each of the wetlands has a different amount of habitats with each different areas. So that was pretty clear. What was as well very significant, like many differences I was able to see at the tidal cycle. That's why I show you this plot here especially, as in a very traditional way, they indeed used intertidal flats during low tide when they were available, and they moved to supertidal habitats at high tide, especially salt pans. And what is more important than this cycle, because this is already very well described in literature, what is more important here is which habitats they use. Like in supertidal habitats, we see many anthropogenic habitats. So we have an artificial coastal lagoon, we have salt pans, we have rice fields that become very important, and that really shows us the importance of various habitats, and even if they are human, they can, in a certain way, represent a refuge for these animals. What was also very interesting in my results was the daily cycle, the day-night cycle, where they did barely not show any significant differences. They basically use the same habitats at day than at night, which is very interesting, even though it's not significant. It shows us that, already as described in literature, and probably, which I can only infer from my results, they are adapted to forage at day and at night, which will leave them then more space to adapt their habitat use across different cycles, such as tidal cycle or the seasonal cycle, without being constrained by daylight to forage. Coming to this most important, most interesting result, in my opinion, the seasonal pattern that revealed hidden habitat importance, being a just like a categorization or like a sub-seasonal view of this habitat use that allowed me to show the importance of certain habitats that would be completely or very largely lost if you had an overall view across the whole cycle. If you just had like one average of the habitat use, you would probably not see that rice field use in the end, for example, during fueling. So that is the major shift towards the end of the season, where they moved away from the classic use of intertidal flats, but used those artificial habitats quite a lot. Now, before I start with the results of the shellfish exploitation activity, I want to give you a little context about it for you to understand and also experience my way of working. I have here the maps that I used, made from data I got about 2 different activities. So here categorized into no activity, low activity, and high activity. So these are densities. The dark red ones describe the 13% I talked about in the beginning. So these are high densities of shellfishes of up to 306 shellfishes per square kilometer. Impressive numbers. That's, as I said, increased, while here it is either plots or no plots in the area Formosa. And the most impressive moment of the last 5 months, I think, was when I started with these GPS locations on top of the satellite data. I already directly was able to see a certain pattern, but what was really interesting is the moment I got this map and I was able to overlay it, as it clearly describes something more important and maybe explains the first picture we got. And I hope you will not find it difficult to understand that these areas, therefore, are shellfish exploitation areas that were significantly avoided by common dredgings. So in this case, we have the activity index for both regions and all classes of shellfish exploitation activity. The dark red ones are the ones of highest activity, highest density of harvesters, or the plots, and both were significantly avoided by dredgings. So that is a functional habitat loss of a key feeding ground for these for the species. And all these results combined clearly showed one thing: that the habitat use of dredgings is influenced by the tidal cycle, by the seasonal cycle, but also by, well, the region, of course, but also the shellfish exploitation. So this is a disturbance that clearly has an impact of the habitat use in a more detailed way, in a more detailed look on the intertidal flats. And it shows the importance of various habitats, which is also my key finding concerning the habitat use in general. They really depend on this mosaic. So lots of habitats that are all connected with each other. There's no such thing as like one key habitat that deserves protection and the other are less important. They are really all important. Even the human ones can be of importance if they are managed in a certain way, as they are, for example, in the Thesis, managed in accordance with the protection measures, like to the benefit of birds as well. And this importance becomes apparent at different moments of the cycles. So what I want you to take home is those 3 messages. First of all, the dredgings do rely on habitat mosaics and not just on one key habitat. Second of all, shellfish exploitation constitutes habitat loss. So habitat loss of, as I said, a key feeding ground, and this within protected areas. So areas that are, amongst others, protected for the sake of birds. And even in the Rio Formosa, this is a legal activity. So legal activity that is planned to increase in the coming years, while in Thesis estuary it's an illegal activity, and despite being illegal, they were able to increase the number of harvesters 70 times fold in the last 15 years. So it is easy to say that the regulation of these protected areas is questionable. And therefore, the most important thing is the implications for conservation and research. So conservation should really, first of all, consider or conserve the suprotidal habitats in the variety as they already exist, but even put more effort into conserving certain habitats such as salt pans in a way that is interesting for shorebirds. But also conservation efforts should aim to regulate and enforce the protection of intertidal flats, which remains the key feeding habitat during low tide. Now, research must consider such variation, so study habitat use variation as I did it, in order to inform these conservation measures that really depend on this very detailed look into that. So, this being said, I want to say thank you to Dr. José Alves and Dr. João Belo, that I think are both watching with the camera. Thank you very much for supervising and for introducing me to the world of birds. It's a fantastic world I was very happy to experience. And as well as the GPS tracking data, tracking analysis work, which was a bit tough to get in first, but it's very useful and I really enjoyed it. And despite this formatting error, I would say thank you to all of you as well. And here are my references. So if you have any questions about these, let me know and we will go back to them. Thank you very much for listening. And
Thank you very much, Emil. Maybe we go the other way. Nico, do you want to start?
Yes, sure. So thank you. I'm all into cycle at the moment, so I'm happy to see that I'm going to be also you did a different way than I do, but that's cool. I have one question specifically about the data that you got for the seasonal cycles. From what I understood, your birds, they migrate a lot in and outside from the two sites you surveyed.
yes.
And I was wondering, then if this migration is seasonal, I guess.
Yes.
How did you, when they're not here, how did you get what kind of habitats they were using?
So I was only focusing on the habitat use of these two areas. So
Okay.
I was not interested in the breeding habitat use. I was really only looking into, into the Thesis estuary and the Ria Formosa.
Okay.
Maybe I was confusing you, sorry if I talked about the whole migration. I just wanted to give the introduction. It is very interesting, and also the research group is also looking into different habitat uses of different areas, including the whole migratory space, so the whole East Atlantic flyway in this case. But I was only looking at these specific habitats.
Okay, but so that means that you have individuals that move in and out
Well,
the region used to be, and then,
they move in and out, but only seasonal. So there's a one migration in autumn that moves in autumn they move southwards, and then in spring they will move upwards again.
But they don't do it. They don't all do it because you still got some for the different part of the year, right?
No. So what I'm talking here about the different seasonal periods. This is within the non-breeding season. So arrival after migration in autumn.
Okay.
Yes. Until fueling, which is pre-migratory in February, March.
Okay.
So this is only describing.
Yeah,
the
that, I guess that would have helped a little bit because when you're not a bird
Yes.
specialist, it's not necessarily super clear.
Yes, thank you.
Okay. Cool. So I guess it was yeah, I don't know. I think that was which came from you. No.
Thank you. I didn't know. I didn't know what was our first shot. What do you have the children?
Sorry?
The chickens, the babies, what do you have them?
When? No, they have them in the north. So the breeding season indeed is happening in the north. So in summer, when the temperatures allow to breed and have their chicks northwards.
So the young ones, they have the chicken is up in the north, and
Yeah.
when they have proper feathers, they migrate with the parents.
Correct. As soon as they're ready, they will migrate with the population. So there's many different exceptions, obviously, as always in ecology. There's all types of patterns and habits, but usually.
For the food, because when I saw this of the rice fields, the first thing I thought, well, so they're eating the seeds. And the reason that they were carnivorous, in fact.
Sorry?
When I saw this thing of the rice
Ye-
fields, I thought that they would be eating the seeds of the rice. But they don't. They are carnivorous, no?
Yes. I don't know if that is 100% the case. I wasn't referring to them as carnivores ever, generalists. But indeed, if you look at them, they do not eat the seeds. They're more interested in different insect larvae, in these areas. That's in fields. I think that's one of the main resources. Otherwise, also larvae of crayfish, for example, which is also happening, also available in rice fields, if I'm not mistaken. So, see.
The worms, snakes, crabs, and clams. What shellfish is this one? Because I always associate shellfish to mussels.
Yes, correct.
But
That is that.
The ones that we get from the south are clams, really, not mussels. But
Different types of mussels and clams. So there's different sorts. As I'm aware, that's mussels, that's clams. In the Ria Formosa, the aquaculture here also is targeting the oysters, which could possibly also be potential prey for retchings.
one of the problems with the farmers is that birds are eating the fish and all that. So farmers are trying to protect the thing of putting nets or something so that the birds cannot go.
Yeah, that's
And the other one is the one that was illegal, in fact, and you found like a million people harvesting there. No? The ones that were illegal, the harvesters.
in the Thesis, yes.
You said that was illegal.
Yes, these activities here are all illegal within the national protected areas, which are here in the black lines. So basically, the whole
One
estuary.
problem is that they are depleting. So they are, one problem may be that they are depleting, that there are no shellfish in that area.
Yes, totally. This is going to be a consequence sooner or later. So far, I can only tell about the direct disturbance of the human activity. Now, according to literature, a certain limit, a certain amount of shellfishes might not be negative yet, like because we're talking about a very huge intertidal area here. But the increase that happened since that literature, since that paper in 2008, is so important that even according to their model, the current density should be very much different, indeed. Also from this regard, especially as here, for example, there's always theories about aquaculture can be positive, and there has been even examples with the common red shank of how aquaculture can be beneficial due to, for example, increased prey availability. That's a theory that exists, but I don't really believe it. I don't think it can be trusted widespread in a widespread way. As you say, they do try obviously to avoid this predation on the product of the aquaculture.
Well, thank you very much. One question. Are the authorities paying attention to your work or
To the legal one, or to the research one?
to the conclusion?
To the conclusions. Well, to mine so far, no. But that is because it's not neither published nor am I established in anything. So that's hopefully to come. But considering the eco-flyway group that I was working with, they are paying attention. There is communication, there is things happening. Now in this area, for example, in the Thesis estuary, they have avoided building an airport in the middle of these salt fans, thanks to this exact research. Yeah.
Thank you.
It's happening.
My turn, I guess.
So,
Thanks a lot for the presentation. Really nice study, and clearly you are showing how the pattern of use. So I think you are not really finding the main reason why. Because one thing that I want to ask is, one thing is to stay in a place, and the other thing is the activity that you are doing in that place. How can you use your data at least to try to infer activity, or because it's not the same the way that you stay, or if you are feeding on the regional, how can you tease apart that, or it's just use in because also depending on the prey biomass availability, is that maybe to get the same amount of food you need to spend longer time. So
These are all very important questions and very important topics that are being studied by the research group. But this is much data that needs to be considered. In my case, it's absolutely not possible. GPS,
I know,
satellite data
but
does not
can you
give any
use
information
part
about
of the data that might be available in the case, so to see before and after the refueling, which is the biomass availability in the different areas that they have?
I was not able to use such data. I know we had them. My supervisor has done himself the mapping of such energy availability in the Thesis estuary. It is still to be done in the Ria Formosa, if I'm not mistaken. So it would only be one region. And these are definitely some layers that are super important to get next in order to get another causal effect really from this.
Yeah, because maybe started up with shorebird, but they correct me if I'm wrong, I'm not an expert on that at all. But they use a stable isotope analysis to really see isotope analysis to really see which which is the main source of energy for them. So to see even before and after the refueling, to see if it changed or not.
I don't know about that one.
Okay.
I mean, this research group
I know
is
it's
not
beyond
as
your
a-
data, of course, but
Yeah.
I was thinking if you can somehow try to correlate or to find some association with your data, with the data of
I know that in this research group, there's, I think, little work about isotopes, but much more about the ecological question of like bioavailability in the such studies. It's trying to correlate it, yeah. But that is happening.
Thanks a lot. Well done.
Thank you.
Yeah. Well, congratulations for the presentation. It was really nice. I like the way you frame all the slides, right amount of text, beautiful images, maps. The way you presented was very, very nice. So congratulations for that. And for the science you did, because it's actually very interesting. I didn't know that these animals can actually keep with their biological rhythms at night. How surprising was that result?
That was not very surprising either. Like together with the title results, these were the least interesting ones because of it, because it was to some point described, but also always in very sites, very far away, in different contexts with different ecological questions. But we did not expect a huge difference between day and night.
They feed at night as.
Because they are adapted. We know they have different foraging techniques, so they're able to probe. If they don't see anything, they will just check out. They will just check if there's something on. During day, they will have more of a less probing, more running, and more visual hunting strategy. So this fits pretty well with that. Yeah.
Nice strategy, nice words. About the habitat use and on the shellfish exploitation banks, do you think you have data not only if there is in a specific place exploitation of clams and so on, but also the amount of exploitation, biomass, maybe the catch per unit effort? Do you have that?
I have the data for the Rio Formosa because as it is legal, it is managed, it is very well. Very well. Because certain bias more or less protocols and what do you say?
Because you have to report that.
Yes, exactly. It's more or less
report the amount of kilos.
So that's existing in Rio, and these are gigantic numbers. Like gigantic numbers, and they try to still increase and increase. They are the number one. Either they are already or they want to become the number one aquaculture area in Portugal for clams and all type of bivalves, especially one species though that makes up, I think, more than half of the all species.
Yeah.
But here it is much more difficult.
Yes.
So the only data we would have here is from scientists like us that do a little bit of probing and sampling here and there. Because there's no such thing. Everything goes into the black market. There's also no regulation here. That becomes a social problem on another side.
Yeah. On that, have you looked at the temporal trends? Because one thing that is affecting the fishermen and could affect actually the birds are flooding events. When salinity drops, many clams and polychaetes and so on cannot cope with that, they die.
Yeah.
And then fishermen cannot obviously get them, but I never thought in the birds. Maybe the birds are affected too.
Totally, that these patterns definitely exist and they are being looked into, but it's very difficult. And the most important thing here would be to actually have a temporal data about the shellfish harvesters, to know their exact movements, to know exactly when there's peaks of activity and when there's none. Because being on the spot, and luckily I was, you can, every time you cross that little bridge here, very big bridge, never mind, you can either see none or plenty of harvesters. It's covered with them. So this is data that would be super interesting, but again, very tricky to get because none of those harvesters is going to be like, yes, of course you can give me a satellite.
You have to put a little back.
We thought about it.
You do with the birds,
We thought about it, yes. No, we were very close to doing it. Really.
Okay, congratulations.
Thank you.
Thank you. Just a few words. I was also very interested, and I think especially the changes, the dynamics of habitat use along the season is super interesting. And I just found a paper that is super interesting regarding the diet. I think Seba and Stop are probably two time-iterative tools to assess that, but you can analyze fecal pellets composition. And especially what's super interesting, that most of the diet is actually made out of insect larvae. And so I think that's why they start, going into the salt pans when it's time to build some fats, to plan for migration, and especially they eat lots of Chironomus salinarius. So I guess it's a species that is mostly specialized on salt pans. So
Can I ask which publication it is? Or
absolutely,
which site?
I can. It was in the marshes of southwestern Spain.
Yes.
Sanchez, Green, and Castellanos in birth in 2005.
And,
You know it?
yeah.
Okay, thank you very much.
Thank you.
And finally,
Not least.
last but not least, I show you the V sign when you have 2 minutes left.
Can I have my
You can have your own timer, but I still show you in case you forgot to look at it.
I'll do that.
So last but not least, Giovanni, whenever you're ready, the floor is yours.
Thank you. Welcome everyone. My name is Giovanni Fetter, and today I'm going to present my thesis entitled Foraging Dynamics of Sympatric Adelie and Ghent penguins across 5 consecutive breeding seasons in Maxwell Bay, Antarctica. To understand the context of this study, first we must look at the Western Antarctic Peninsula. This is one of the most fastest warming places on Earth. The regional warming is due mainly to anthropogenic reasons, and these environmental shifts can trigger biotic responses in species that are sensitive to climate change, altering their behavior, their population dynamics, or even their physiology. This is the case for 2 pygoscilid penguin species like Adelie and Ghent penguins. Specifically, I'm focusing on Ardley Island, which is a protected natural area located in Maxwell Bay, which is located as well in King George Island in the Western Antarctic Peninsula. Here, there is a sympatric breeding colony where these 2 species nest side by side. Interestingly, while they target the same marine prey, which is Antarctic krill, they are showing divergent localized population trends, with Adelie species decreasing over time, while Ghentus are constantly expanding. They also show species-specific strategies to target this prey, and these strategies can be also modulated by oceanographic drivers. Between these oceanographic drivers, tides emerge as one of the most important ones, affecting prey availability. With this in mind, this study focused on 3 main dimensions, each one associated with one specific hypothesis. The first one is related to vertical use, where it was hypothesized that species will show differential vertical range utilization across the marine depth strata, where Ghentus, due to their larger morphological differences, will exploit deeper and even benthic layers of the water column, while Adelies will focus on the more pelagic, shallower layers. The second one is related to the horizontal views, where it was hypothesized that species will exhibit distinct spatial segregation and distinct habitat utilization. Finally, related to the oceanographic drivers, it was hypothesized that tidal phases of tidal cycles will alter foraging, will modulate foraging intensity and hotspots location in the bay. To evaluate for this hypothesis, we deployed multisensorial data loggers on the lower back of breeding adult penguins across 5 consecutive breeding seasons. So from 2019-2020 to 2023-2024. These loggers record GPS location as well as temperature and depth and 3D accelerometry. They were left on the individuals for 3 to 7 days, and then once they were retrieved, the data was filtered in order to eliminate incomplete trips or records obtained from devices that were damaged. For this study, we're focusing only on foraging dives. So all the other dives, like recreational or transit dives, needed to be excluded. To do this, we actually focus on the accelerometry information. So we got, for example, with 1 second interval from any dive. We fit like a marine learning algorithm that actually classifies the behavioral conduct of the penguin in each dive. So if 1 second of the dive was characterized by the model with more than 50% of 1 second was characterized as a capture prey attempt, then that will be like characterized as a capture second, foraging second. But with this in mind, we got like, for example, one dive that lasts 30 seconds. If at least 5 or more seconds were foraging seconds, then this would be considered a foraging dive. And we only analyzed these foraging dives. Moreover, we also integrated environmental features like bathymetry and tidal cycles. For bathymetry, global models can frequently smooth out like critical coastal features like inner fjords, like in the bay. To correct for this, we actually joined together the 2026 global GEPCO grid with high-resolution bathymetric surveys provided by the Chilean Navy. With this, we obtained a high-fidelity bathymetric model for the bay. Simultaneously, we also developed a synthetic tidal model. To do this, we actually obtained sea level records from a local station that was active in the area in the 90s. We decomposed its signal into its 8 main constituents, and with this in mind, we projected a synthetic time series, sea level time series, until 2026, which was then, of course, validated and cross-compared with official tide tables from the Antarctic bases in this island, with high predictive accuracy. After all this long process, and after all many years of study, we got a high-resolution dataset that obtained information from 72 adult individuals, 85 Ghent penguins, with dives from each species, from more than 50,000 dives for each species, comprising a total amount of more than 120,000 individual dives that provided us critical information like diving duration, diving depth, location, height above the seafloor, tidal stage, among others. With all this, we conducted several statistical analyses. The first one was a generalized linear mixed model to account for differences in diving depth between species and across seasons. This model showed significant differences, where Gentus showed a mean marginal diving depth of around 48.8 meters, which represents a 43% increase compared to the shallower 34.2 meters that Adelie usually dive to. Moreover, our results across seasons show a parallel dive pattern, where both species follow the same pattern across the seasons. Interestingly, in 2021-2022 season, which was considered as a low creel availability season, both species show a significant deeper diving, which we can see here, which can also represent an increase in vertical search effort for these species. We continue our analysis with water column utilization. So basically, their diving depth compared to the seafloor, to the bathymetry. Here, Adelie show like internal shifts compared to the Gentus that show more strict stability. But I want to focus on this plot because it can be tricky. First of all, the y-axis is inverted to common oceanographic plots. As the 0, here it represents the sea level instead of the sea surface. So for example, for Adelie here in 2019-2020, we can see that the bar is concentrated on the lower part. This doesn't mean that here we found Adelie penguins foraging at 2,000 meters. That would be a shocking finding, and it's not possible. What this represents is that these Adelie penguins were foraging close to the seafloor in shallower areas of the bay. This is also due because in this year, it was actually a high availability creel year in this area. So to save effort and save energetic demands, penguins forage close to the to the coastal features of the area. Nonetheless, on the next 3 years, their krill availability decreased, forcing them to expand their foraging grounds into deeper bathymetric features, while with this naturally inflating their variance of height above the seafloor. For the gentus, they showed no significant difference in this height above the seafloor, so they consisted part interannual pattern in this distribution, which can be associated with another thing that we analyzed, that was benthic foraging. We analyzed first like raw data, and out of all their dives, for gentus, 12% were benthic, compared to just 5% from Adelis. But when implementing our binomial generalized linear mixed model, we actually found out a higher probability, and even higher than this probability, of almost 4 times higher, or more likely to conduct benthic foraging. This can be related, as I mentioned in the hypothesis, to morphological constraints. Gentoo penguins are larger and have a higher aerobic diving limit, which allows them to stay underwater for longer, targeting these benthic layers that can also help to mechanically trap the prey and the krill using the seafloor, which allows them to act as an ecological buffer, allowing them to decouple from the environmental fluctuation in the upper layer of the water column. Now moving on to our horizontal spatial analysis. To quantify horizontal overlap, we used the Batacarias affinity index, which is a simple index that ranges from 0 to 1, where 0 means complete segregation and 1 means complete overlap. So when analyzing for general results, the overlap was moderate to high of 0.741. But when we deconstruct this by habitat type, meaning pelagic and benthic, we found some interesting results. So for the pelagic habitat, the overlap remained high, even higher than before, which means that this is because two species, the two individuals from different species, can actually be on the same geographical location, but exploiting different vertical niches. So they avoiding direct interference. Nevertheless, when we just look at the benthic habitat, they cannot do this. So they mediate this by finding different habitats and with intensified segregation in this use. And we conducted this Batacarias affinity index using utilization distributions, which are maps like this that plot foraging intensity probability. Now, finally, introducing to our analysis the oceanographic drivers and tides. Here, instead of comparing one species against the other, what we actually did was compare the same species between tidal phases. So, for example, Adelis during their foraging grounds during low tide with Adelis and their foraging ground in high tide and ebb tide and in flood tide. We did this in two dimensions, which we call general home range and core areas. So basically, we plotted also maps like this, utilization distributions, and the foraging perimeter we considered for home range 95%, and then we just focus on the 50% inner core area. Here we have also some interesting results. So for both species, the general home range did not change. Their overlap was high, which means that they basically forage in the same areas, no matter the tidal stage. Nonetheless, when we just focus on the core areas, we have differences in species. So Adelis showed a minimum overlap index of 0.62, which is less than before, but they still share forage in the same areas across tidal phases. Nonetheless, Gentoo show a significant difference, not significant, but a difference of minimum overlap index of 0.2. This is that they use fine scale localization praise across tidal phases that can be due, and we suggest that it's because of mechanical aggregation of krill during entering tides or high tides, etc. To conclude, this 5-year study suggests that the coexistence between these two pygoselids can be mediated by by species-specific behavioral flexibility rather than ecological boundaries. There is not like a limit or a physical barrier that segregates them. Nonetheless, as shown in these three pillars that I discussed throughout this presentation, the foraging dynamics of both penguin species are mediated by robust ecological patterns, large-scale patterns, rather than just individual penguin decisions of where to hunt. In Western Antarctic Peninsula that's facing unprecedented climate change, it is important to understand how these robust ecological patterns affect the resilience of each species in the area. So we can apply and introduce marine conservation strategies that help us protect and save changing Antarctica. Thank you very much for your attention, as well as I want to thank my supervisors, Lorena, Alvaro, my friend's family, and the IMBRC Consortium, and the Chilean and Uruguayan Antarctic Institute. Thank you.
Thank you, Giovanni. Now we all hate you here. It was not a very wise move.
I hope they didn't affect my grade.
Who wants to start? Not Nico, but any other name. Yeah.
Can you go back to the figure with the how many dives the different species of that one?
Yes.
You said the little ones are 72 instead of 85, and yet they have many more dives.
Yes.
Can you explain that? Because they have to use a lot of energy for the dive itself, no?
Yes, when this was conducted through several seasons, so it's actually like previously recorded also in literature that these are like more dynamic prey species. So when they go out in these foraging trips, they conduct fast and continuous simultaneous dives, so they can return like as quickly as possible to feed their chicks. While gentus have been shown to not be done consistently, they are not as in a rush as these other species. That's why the difference.
Why? Maybe the bigger ones have someone else taking care of the chickens, or do they have? Because that looks like awfully expensive for the little ones to be dying so much.
Yeah, and actually they are also really mindful of how they expend their energy. They are tracking the environmental conditions. So if prey is moved from the coastal to other more open areas, they will track this prey. Where gentus mainly are not showing this behavior, and maybe that's one of the reasons that we need to study why this species is decreasing. Could be a reason.
Then I comment that it is a pity because the most visual and important information that you have, it is actually from here I cannot see. Those figures, the figure that you have there,
yes,
I cannot see what it is. And the next one I cannot see. It's impossible to read.
Okay, I can explain it, and next time I will be a bigger picture.
And the next one, the same, you should have put that much bigger.
Yep.
And the numbers, in the middle of Thank you.
Yes.
I agree with this thing of the figure. However, you did a great job guiding us through the whole analysis, so that it's tons of work. So in that sense, yeah, of course, I would suggest you to increase this because it's difficult. I mean, we are getting older, we need glasses, so yeah.
I did not see your thesis.
Yeah. So, but this, I think you did a great summary of a huge amount of data. So in that sense, congrats on that huge effort that you did. So I have more like a general question. I think, correct me if I'm wrong, but you say Adelaide penguins, they are decreasing in terms of the population?
They are decreasing in this specific colony. Yeah, actually.
However, they show much more flexibility, no? At least in changing the depth at different time of the year, and also the use. So the usage seems to be differential, at least in the vertical, no?
Yes.
So, what do you think are the main drivers, at least in that population that you study, of the decline of these Adelaide penguins?
Yes, that's a very interesting question that all the research group that I work with is asking, because there are like several factors that could be interfering with this, affecting this decline in the population. We are not sure if krill availability or like foraging dynamics is one of these factors affecting it, because I mean, even though the interannual variability of krill could be varying, and some years could have more or less, there is supposed to be enough krill to maintain the number of species, the number of individuals and species there. So maybe it could be related with other macroenvironmental factors like climate change. Temperature is rising in this area with more constant heat waves. There is more like boats and krill fisheries, as well as some oceanographic features that could be interesting, like the intrusion of warm water in the bay that coming from the Bransfield Strait. So this is just part of a bigger study that is trying to answer that question of why are they decreasing.
Okay. Yeah, and related with that, I'm sure this is part of a huge study. So, which part of yours, all this was done by you? Of course, I guess all the analysis, but
yeah, the analysis. I was doing field work on this past season, which the data was not. Yeah, so and then the identification of foraging dives. This was conducted by one of my supervisors, where she classified this with a software that's called Igor Pro, using and analyzing all the 3D accelerometry, which is very complicated work. So I already got like a data set with all the records from the dives, with like individual ID penguin with each dive, the depth, and then I could analyze it and compare it with the bathymetry and tidal model, which I developed from scratch for this thesis.
Yeah, I really like the way that you explain, for at least for non-expert like me, for example. So what that was really great.
Thank you very much.
Nico.
Nico.
I guess I can go. Yeah, I really appreciated your talk. Some few things, but my colleagues already said that maybe perhaps the figures a bit bigger sometimes, but otherwise, yeah, it was really well guided. And yeah, I really appreciated that generally. I have a very general question on the ecology, and on one of the main results of the study is that there is some spatial separation between the two species, and you interpret that as a competition avoiding mechanism, but still they eat on the same food, so we can imagine that they deplete the same stock anyway. So mechanistically, how does it work, this competition avoidance?
Yeah, that's also an interesting debate that we had with my supervisor, if it's actually like a competition. Like, for example, the Gauss principle of competition exclusion, that states that two species that habitat the same place and target the same resources, if the resources are limited, one of them will outcompete the other. And so that's interesting how this is applied here. And what we face is that mechanisms like this segregation in the pelagic layer or benthic habitat, as well as their different habitat distributions, could mediate this competition, so it's not doesn't result in one species outcompeting the other. So it's not like they are directly competing between one another, because they have these mechanisms to attenuate that.
But how if they eat the same? If they deplete the same stock, if the resource goes down for the resource will go down for both of them, right?
I'm sorry?
If they deplete the same stock,
yeah.
If we consider the same crystal, the resource will go down for both of the species, right?
Yes, but then they utilize different. For example, they are foraging in the same pelagic layer on the shallower column, so they reserve energy. If the resource start to decrease, then, for example, Gentus could apply other strategies like this benthic foraging, where they find prey in deeper layers, or Adelis could expand to more deeper open areas. And that way, even if the prey is decreasing in the general shallower column, they can still both find prey.
Oh yeah, I had one about the machine learning part.
Okay.
I don't know if you did it yourself.
No, but I can try to answer.
Okay.
Still.
I was a bit curious because you need to train those models.
Yeah.
And so this one was trained to find this part of the signal is hunt. This one is not hunt or travel or whatever.
Yes.
How do you, how were the model, those models trained? How do you annotate this kind of data to move it into the model?
I'm not sure how it was trained because it was from previous years and it was conducted by other researchers. But what I can tell is that according to the movement of the accelerometry in the 3 dimensions, it classifies it as capture attempt, rest, traveling, and there's another extra. And I'm not sure how it's trained, but I know that, for example, to verify that it's accurate model, what we are doing now, we started this last season, is also attaching cameras to the back of the penguins, and then comparing the videos of them and of their activity, and to see if that aligns with what the accelerometry is telling us. So if we see in a video that they are like there's head movement and they try to eat some krill, we can overlap it with the accelerometry and see if in that exact same time frame it was registered by the accelerometry and by the machine learning models. If that was also a capture attempt, and the test trials are like positive and it's matching.
Nico? Okay. Well, thank you very much for this presentation. It was really nice, very easy to follow. Very nice science you present here. Thank you very much for that. I have a curiosity. I think in the now you didn't mention that, but in the presentation, I think you mentioned another category of behavior that was recreational diving. Did you say that?
Yeah.
So they do that or what?
Yeah, especially for example for fun in like we see in the movies and series in small icebergs, and you can see them like just going up, then down, diving a bit, and going like back into the iceberg. And from what I discussed with my supervisors, they're just spending some time.
Yeah, I mean, but is this a category that is called other kind of behavior that we don't know?
Yeah.
Yeah.
Not
than
recreational,
recreational, right?
Yeah, no.
More
That's
than
just for
I'm going to have a swim and see my friends.
No.
Okay. And on the other hand, did you have the opportunity to cross this data with CTD profiles or vertical profiles of temperature? Because you mentioned the heat waves.
Yes.
Heat waves there are, as far as I know, they are in the mid-water, deep waters. That is the warm water center at a certain depth. There.
Yes.
So you should see an inverse thermocline. Warmer waters, deep.
Under, yeah, deeper.
Have you the chance?
Actually, the first proposal of this thesis was actually to analyze both oceanographic variability, especially with thermal stress and fluorescence. With this diving, we actually plan to deploy an oceanographic LiDAR at the same time as the loggers to see like the whole dynamic of the bay. But in the end, it couldn't be possible. But it's been discussed. There's like biologgers that have like small CTDs that we could be attached to the penguins, and then we had like the perfect match. Yeah.
So this was not possible this year, last year, but not because you mentioned that you have 5
Yeah,
years.
no,
it was not possible any year.
No.
Okay, thank you very much.
Thank you.
Thank you very much.
Thank you.
Maybe just a few concluding words. We've seen 3 great presentations in this final session, but beyond those 3 presentations, I think I can speak in the name of all my colleagues to say that we've seen many great presentations, and I think you can be very proud of yourself. We are proud of you, and I consider as a teacher, it's one of my greatest privilege of my job to witness that. We take baby students when they enter the master, and then they open their wings, hopefully not lucky. penguin in a more efficient way. And yeah, it's always something very important, and it's yeah, great privilege of our life. So I'm going to